Hi, everyone. I'm Christina, and I'm so happy to see you all here. So I think that it's time to start. And before starting, I just wanted to highlight the logo. I don't know if you've noticed it. It's scale. You see the N-acetyl aspartate, and that's a hallmark of our community, the MRS community. And it's also a hallmark of Switzerland, the watch. And it was designed by Dunia, who's there. So it was designed a few weeks before she had her PhD defense. So yeah, that's an accomplishment. So I would like to, so the talk of today, it will be fast. It will be divided in two parts, a welcome and a scientific introduction. And first, I would like to, as I said, welcome you all here in Switzerland, the country of cheese, the country of chocolate. 2 days ago, I found it out again and again that the milk chocolate was invented here in Switzerland in 1800s by Caillet. So yeah, something to know. So thank you for being here. You're 135 people on site and another 35 to 40 people online. So thank you again. And thank you for the speakers, for all the poster presenters. Thank you for everyone who played the game, who came and submitted their work. So I hope that you know that without you, this workshop wouldn't have been such a big success. It's already a big success, yeah. So before going further and thanking other people, I just want to hover the past workshops, the ones that I remember because I'm a very young scientist. So the one from 2016 was organized by ISMRM here in Germany. The one from 2018 was organized in Utrecht, and the one from now, which is organized here at EPFL. And I would just like to highlight the link between the governing committee from the MRS study group at ISMRM. It's a tradition that it was installed over the year, and I really hope that this will continue going, that the workshops will be organized with the spectroscopy study group at ISMRM. So now you're probably wondering who are these amazing, very young scientists. So because the aim of the workshop of today is to bring the people together, to bring the young scientists together with the more senior ones, because of the past, the lack of meetings in the past. So these are quite young. I hope that you agree. And they were like you some time ago. So the challenge is to recognize them here in the room. A hint, they are the scientific organizing committee, and here is their names. So I would like to thank them. They are extraordinary people. They were amazing in organizing this meeting, this workshop. So thank you very much. I'm going now with a big thank you to the local organizing committee. They are my colleagues from CIBM. They are also amazing. And yeah, I just want you to know them. You saw them at the registration desk. So they are Bernard, Sarah, Dunia, Jesse, Jan, Brian, Florian, Kasia, Daniel, Lijing, Antoine, and Hélène. So thank you very much for this. Without you, we wouldn't be here, or I would be going crazy here. So you all notice that the workshop is organized at EPFL. So EPFL is hosting the workshop, and we have here the Associate Vice President from Centers and Platforms. CIBM is a center, a research center, so it's Professor Anna Fontcuberta, who will be with us today. And we also have the Dean from the Basic Science School, Professor Paul Dyson, here today with us. So thank you for allowing us to have the meeting here. The workshop is also supported by the CIBM. So today with us, we have the CIBM Executive Director, Dr. Pina Mazziliano, who's there, yes. And we are also supported by the Swiss National Science Foundation. And we have Nestle, Newsoft, Mendes, Bruker, and Rapid as sponsors, as donators. And they here allowed us to provide the travel awards, the travel stipends for you to be able to travel, meet us. So, as you probably know, Switzerland is the neutral ground. It's a ground for negotiations. So we have a big room in case that the meeting goes wide. So like this, you're apart, not because of COVID, just because of the fact that you're a neutral country. But in case that you get bored, we have a second option. So we have the media experts and we have a game. So now I would like to welcome Dunia here. She will talk a little bit about the media experts, and then Jesse will talk a little bit about the game.
Hello, everyone. So first, I would like to say a big thanks to the scientific organizing committee who supported this idea from the start and included it in the program. So, and also like to organize my fellow students who helped with all the backup and organization of the Meet the Experts session. I would like to thank all the experts. We have 44 experts participating, so a big thank you to you all. And I would like to explain a little bit. So in every coffee break we have, and the end of the lunch break, you all received your schedule. We have 8 expert tables with 2 to 3 experts. And the student groups of 7 to 8 trainees are assigned to each table. And in each break, the students will shuffle. So as many students meet as many experts, that's the point. I hope now I'm counting on the students for you to use this opportunity to meet the experts, make new acquaintances, and enjoy it and just have fun. So thank you very much.
So yes, hello everyone. So we, the local organizing committee, decided to introduce a small game for the workshop. So we asked every oral presenter to hide a small cow in one of their 5 last slides. And the challenge for you is to find the location of the cow. So you need to identify in every speaker's presentation the position of the cow. And so this is the cow we sent to the speakers. So this is, she's called Happy Lily. It's an actual emblem of Swiss tourism. So it's, yeah, very local. So you see, we send this few designs, but the speakers could change the colors as they wanted, not the form. And as an example, I sent a slide. This is one of my acknowledgement slide, and I added Lily here in as part of Christina's team. So yeah, that was an example, but I think the speakers were much more creative than this. So be attentive. This is the people that started the idea. So thank you, Mladen, if you're around, and Roland for continuing the idea, and for everyone who helped organizing the challenge. So you got at the back of your badge a small answer sheet. So you have to put your name, tick this box if you don't see the colors. And so for each presenter, you have to input the slide number where you could find the cow, the position on the slide, so top left, top right, center, bottom left, bottom right, and the color. And for the color, we had a small discussion last night. So let's decide that the color would be the color of the contour of the cow, not of the spots. So for this example, the color would be black. And if it's none of this color, you just tick other. And so the answer sheets will be collected on Wednesday noon. So sorry for the last speakers who are not part of didn't get the chance to put the cow in their presentations. And the results will be announced on Wednesday afternoon after the last speaker. And we will have 3 small prize for the winners. I hope you enjoyed this activity. And if you have any question, if it's not clear, just yes. You have to guess. I think, yeah, Christina has a cow.
I have a cow, but I did not put it in the last. I was too stressed. Let's putting it like this. It was on the first slide, my cow. So I will go back to my first slide at the end, and yeah.
Okay, so starting from now, please let's try not to cheat. And yes. So yes, all clear. Thank you.
I'm not done, huh? So posters, we have 42 on-site posters. You have the section there when they will be, but feel free to go and explore them. They were installed this morning, and you all received the Friday at noon, a Google Drive with the proceedings, so you can have the abstracts for all the posters there. We also have 8 online posters. They are also hanged here, but as an A3 format. You have to connect on Discord. You have a QR code. It's provided also on the proceedings, and then you'll have a chat window to discuss with the at a specific moment if you have questions with the first author of each of the posters. We also have some social events. So we are welcome you Tuesday evening to the Olympic Museum. We will be leaving from here at around 5 after the session. There will be groups of people leaving from here, or you can go directly. You have also the instructions in the proceedings. And then this evening, the trainings organize a drink and a dinner at the Great Escape. It looks like this, just to show you that it's an amazing place. It's an amazing place for people to meet, to have a burger and discuss. Hope that everybody will join, and yeah. Then you probably know we have a special issue in NMI and biomedicine. We are, how can I say, we are welcoming original contributions, so new research articles, not reviews. And they have to be submitted as a regular manuscript, and they will go through reviewing and so on. The deadlines of the submission will start in December 2022, and it will end in May 2023. And here are your handling editors, Peter, Wolfgang, Christina, Christoph, and Georg. So for emergency, you also have some information. You have the campus application, you have a phone here. And for those who need a COVID test to go back to their home countries, you can also organize one. You have a link here under the proceedings. So you have to take an appointment, and it's quite close by on the EPFL campus, so you can get tested. So now that you've seen all the people who worked very hard to get this workshop done, and you know what to do in case of an emergency, so really run like hell. Now let's go to the scientific introduction. So I'm starting my scientific introduction with a small overview of the workshop of 2018. As I said, I like to see them as a continuation, and especially because they organized always with the MRS study group at ISMRM. And the first talk was given by Arend. Arend is here with us, but online. And he made an amazing overview, historical overview of spectroscopy, starting with 1970 with the work of Peter Mansfield, with the work of Paul Lauterberg. And there were two things that draw my attention. The fact in the 1985, the researchers at that time believed that all the MRI problems were solved. However, they were disappointed by the fact that the biological information was not so strong. So then they had an amazing idea, go to MR spectroscopy. So they did the first MR spectra from phosphorus. I hope that Steve agrees with me because it's in front here. And they were very inventive. So I hope that we will continue to be inventive, and we will continue working together. However, Arend also highlighted the fact that we need to move even closer to the clinical arena. We need to go even faster there. And he provided some ideas like the need of killer biological application, like discovery of new metabolites, like the 2-hydroxyglutarate, or new roles or new changes of metabolites like GABA, glutamine, and glutamate. He was talking about energy metabolism, who's quite important in oncology, using deuterium imaging, MRSI of prostate who advanced very, very fast, and also the role of MRS in brain function. He also highlighted some killer developments like MR fingerprinting. At that time was widely used in MRI, started to be used in phosphorus MRS, the deep learnings, the MRSI, and other techniques who needed to be faster, more robust, and more automatic way of data processing. So I would like to somehow go to the program now and show you that we are working on it, and that there are some very interesting talks that you should be aware of. Then in addition, he also said that there is a need of consensus on methods and applications, multi-center, multi-vendor studies, harmonization, and also an important translational research who has to be still continued. So because we talk about people working together, about community, in 2019, the MRSHub was born. So this was after the workshop in 2018. So it's a new thing. I really hope that everybody knows it. If you don't know it, now you know it. And it contains a collection of resources for analysis of MRS data, but also contains an amazing forum. You have a question, somebody is there always to answer. And this somebody, it's sometimes Georg, who's here, will have the first talk. It can be Candace, it can be Will, or all of you. And this is amazing. I've posted a question 2 days ago. I had my answer in 6 hours, I think. So it contains also a huge amount of educational materials related to MRS that Candice was here. She organized them in a very fast way, and she will be talking about it later on today. we were talking about the consensus papers. So as you know, they're out. So there are 13 expert consensus recommendations who are out in 2020. So this is an amazing work showing a lot of progress and advancements in MRS, MRSI. And we will be talking about these consensus papers on Wednesday. Then killer applications. We will be talking about metabolites today in the morning. We will be talking about X nuclei on Wednesday. And we will be also having a session on clinical applications. What more developments? MR fingerprinting. We have Asaf here with us today, and he will be talking about it. Deep learning, we have a session on it, denoising, and so on. So we are covering all these fields, and we are continuing to be inventive, which is quite amazing. Killer developments, fast MRSI, we have a session on it. We have even a panel discussion, multi-center, multi-vendor studies, harmonization. We also have the session number 6, data processing, the session number 4. So I really hope that I've convinced you that we are working on it. You also have to know that for the panel discussion on Wednesday on MRSI, we have a bingo, so we will be voting. So you will have a link with you have the link here, you will have a number where you will be connecting. And with your phone connected to the internet, there is a free internet here, you'll be able to vote. Then, in addition to this, the multi-center, multi-vendor studies are also in the session 6 highlighted. And the need of translational research, which is very, very important, it's highlighted in the preclinical session. However, this year I gave a talk at an educational talk at ISMRM, and trying to prepare my talk, I realized that there is something missing in the preclinical MRS community. 10 years ago, 20 years ago, the preclinical MRS community was wide, was working hard. There were a lot of abstracts submitted ISMRM, a lot of sessions. And the clinical one was slightly after, following it. But in the meantime, things changed. The clinical community now is really, really on top, really having amazing studying, amazing results. They did their part of the job, bringing MRS in the clinical arena. However, I felt that we need to provide a starting point for a standardized protocol and instructions for new sites to follow for the preclinical experiment. So we were discussing with Jamie Neer, and we would like to propose a multi-center preclinical MRS study, try to do similar things which were done on the clinical spectroscopy community, and bringing people together, showing that we can have reproducible results in the preclinical arena. We would like to assess within sites and between sites variability of metabolites measured using standardized rodent brain MRS experiments. First idea using ultra-high fields, multi-vendor, Bruker, Varian, and so on. Trying to find a common brain regions, a positioning and anesthesia, common acquisition parameters, or other acquisition parameters. Data processing and analysis will be done. So some steps will be provided and others will be left free. We would like also to compare sets side by side versus single centralized processing quantification. I hope that people are interested in this type of study, and I hope that you will be looking forward to work with us, and we are looking for contributors. Of course, we will be following the guidelines from the consensus paper, which is here. And I just posted this morning this on the MRSHub about this preclinical study. I hope that we will have answers and people contributing. So now I would like to thank you for being here one more time. And I hope that you have fun. So don't forget, fun is very important. And I don't know if Pina would like to say a few words. And then we will pass to the session number 1.
Is it on? Yeah. Okay. So I'll be very brief. Just want to give a very big thanks to Christina, because without Christina, this will not be happening. And I would like to welcome you all and enjoy the event. And I hope many intellectual stimulating discussions take place. And that's it. Thank you.
a senior moderator and a young scientist moderator. So we'll be like this all the sessions to try to open the discussion.
Okay, thank you very much, Christina. Thank you for the organization. I'm extremely happy to see that spectroscopy attracts a lot of people. As you heard and as you know, spectroscopy has a long history and a lot of promise. And by the way, never come really in a real diagnostic arena. And I hope that this 3 days workshop will help to understand why and help to find ways to bring this wonderful method into the clinics. So I introduce myself. My name is François Lazera. I'm working at the CIBM in Geneva. And I will co-chair this session with Gianna Mossa, from Purdue University in Indiana, US. So I have seen that there is a lot of people that made the travels across the Atlantic Ocean, and I welcome them. And I hope that they will be able to enjoy Switzerland and the region of Lausanne after 3 days of spectroscopy, looking at little peaks. They will enjoy to see larger peaks around the Alps. So I don't want to to spend more time.
Keep going.
The timing is sharp. And I have the pleasure to welcome the first speaker. And the first 3 speakers will be on basics science and basic spectroscopy. And the first one will be given by George Uxner. I hope I don't mispronounce your name too badly, from Johns Hopkins Hospital. So please, the floor is yours.
Thanks, Francois. Is that working for everyone, microphone-wise? Wonderful. Well, yeah, let me also say thank you very much, particularly to the organizers. This is probably the biggest talk I've given so far. This is a huge, huge, huge honor for opening this superb workshop. And I hope I will manage to make an honest attempt to bringing us all to speed on basics of single box spectroscopy and MRSI in about 20 minutes. So let's see how that goes. So I wanted to start with a bit of a raison d'etre. So the question is, why do we want to localize spectroscopy signals in the first place? Well, first of all, of course, we want to be able to make inferences about the spatial distribution of metabolites. But in many cases, localization is really what allows us to obtain usable data in the first place, because we can avoid signals that would otherwise overwhelm our spectra, like the extracranial lipids in the brain, for example. And obviously, optimizing field homogeneity is easier over a small and well-defined volume, and that will give us narrower lines and better signal excitation. You're right? Okay. And it helps with water suppression as well. So I wanted to first talk about a couple of minutes about single box spectroscopy. I wanted to review just a few basic shared concepts, look at the pros and cons of the most widely used sequences, which I think is quite useful. And we'll go through a couple of practical considerations too. So just to go back a little, you will probably recall that slice selection MRI is typically achieved by combining a field gradient that temporarily makes the Larmor frequency a function of a spatial coordinate, and a frequency selective radio frequency pulse that acts on a whole range of frequencies, and in space that translates to a slice. And pretty much all single voxel sequences really work by just consecutively selecting 3 orthogonal slices. And the goal is ultimately to select signal from the intersection of these slices and suppress signal from everywhere else. And before we look at the most common ways of achieving that, I wanted to offer you a quick refresher on like 3 important concepts, and they will help us understand the pros and cons of each sequence. The first concept was the RF pulse bandwidth, just kind of the range of frequencies that a pulse acts on as intended. And for any particular given pulse shape, there's an inverse relationship between the bandwidth and the duration of the pulse. The second thing to know about bandwidth, without going too deep into the details, so-called adiabatic pulses have considerably larger bandwidths than conventional amplitude modulated pulses. And that concept has important implications for this phenomenon that we call chemical shift displacement effect, since we just record that different Larmor frequencies during slice selection will be excited in different slices. But we also know, excuse me, that different peaks in our spectrum have different Larmor frequencies as well. So different peaks will be excited in different slices, which is obviously something that is not necessarily desirable. But it turns out that high bandwidth pulses are pretty much the only way to mitigate that effect. The final concept is the specific absorption rate, or SAR. We know that radio frequency pulses heat tissue. SAR is a measurement of that power deposition, and there are very strict legal limits to ensure that scanning is safe for participants. SAR is interestingly proportional to the square of the magnetic field strength, for example, the square of the flip angle as well, to the total amount of time during the sequence that you spend irradiating radio frequency pulses. So if you go to higher field strength, you're using a lot of high flip angle pulses, SAR will be increased. And generally, what we will find is that sequence design typically requires some sort of trade-off. So we can't really design something that's perfect. So let's talk about the strengths and the weaknesses that these concepts bring about with them. And I wanted to start with press because that's pretty much probably the sequence that everyone in this room, I would imagine, has used at some point in time. And press uses amplitude modulated slice selective pulses, a 90-degree pulse for excitation, 2 180 pulses for refocusing. And that's a really easily implementable way of generating a spin echo. And you can find press pretty much on every clinical scanner. But it turns out that those pulses are also press's own worst enemy, so to say. They are quite low bandwidth, right? That causes a large chemical shift displacement artifact to 35% at 3 Tesla. That also reduces sensitivity for coupled metabolites, through something that we call destructive J modulation. The 4-compartment artifacts, for example, things like lactate are a bit more difficult to detect with press. The 180 pulses also eat up a lot of time. So the lowest possible echo time you can achieve around 30 milliseconds. You lose a lot of signal to T2 decay. And these pulses are very sensitive to being to the B1 being miscalibrated or the RF delivery being inhomogeneous. And that all may be somewhat tolerable at maybe 1.5 or 3T, but these issues really become quickly unacceptable at higher field. Spatial is indeed special because unlike any of the other sequences we're talking about, it requires 2 shots for localization. And the basic module is still pretty much 2/3 of press. So it's a 90 and a 180 to generate signal in a slab. And in the third slice selection direction, you use a frequency selective inversion pulse in the first of 2 shots. And when you subtract these 2 shots from each other, the signal outside this pre-inverted slice is subtracted out because it's identical, and all that's left is that slice. And together with the slab from the 90 and 180, you get the localization. So why do that? Well, the advantage is by removing one of the 180 pulses after excitation, Spectral can go to very short echo times. It's well recommended to do that. The downside, obviously, is you're using 2 shots for localization, and you rely on everything that you don't want in there to cancel out perfectly. And that can be particularly tricky with things like lipids, but also subject motion is an issue that becomes difficult with Spectral sometimes. Chemical shift displacement is still quite high because the pulses are still low bandwidth. And frankly, Spectral is also just not very widely available. Steam uses a different way to generate a signal. So instead of a spin echo, Steam selects a so-called stimulated echo. And it does that with 3 size-selective 90-degree pulses, which are obviously shorter than 180s. And that's quite good for several reasons. They all fit into a sequence that is quite short, up to a few milliseconds. Of course, they're short. They also have a higher bandwidth. So chemical shift displacement is typically lower. They have low SAR. And Steam is also still available on pretty much every system. So there's a lot of advantages, but one major drawback that Steam has, the stimulated echo that we're detecting, is only half the intensity of a spin echo. So you're quickly running out of signal. And that may be a deal breaker for a lot of applications. But particularly at 7 Tesla, the advantages of Steam still play pretty well, particularly if you want to go to short echo times. So it's still pretty widely used. For the advantages of adiabatic pulses, let's recall they have very high bandwidth. So they bring us really very low chemical shift displacement, good co-localization. And they're inherently quite insensitive to B1 variation, B1 inhomogeneity. And the laser sequence really takes full advantage of that to maximize kind of location fidelity, if we want to say that. And it starts with a non-selective 90 for excitation and even 3 pairs of slice selective 180s to get localization that's as good as anything you'll get. But you kind of see the problem. It's a lot of pulses. Adiabatic pulses need to come in pairs anyway. It's introduced like a little phase lag that needs rewinding. It also means our minimum echo time is long. SAR is quite high. There's little time to do anything else. And also laser is not really readily available on many systems. And now ultimately semi-laser then is an attempt to have our cake and eat it and to kind of compile a best of almost. So it combines the excitation module from press with the adiabatic refocusing module from laser. And so we obtain very low chemical shift displacement in the refocusing directions. There's still some in the excitation direction, but it's a 90. That's quite acceptable bandwidth still. It's largely robust to B1 homogeneity, which is a particular issue during refocusing. Doesn't allow for an echo time quite as short as steam, for example. SAR is higher too. But the overall package is so well balanced that semi-laser has earned the expert consensus recommendation for most applications. And there's also now been an effort to deliver a cross-vendor standardized version of it to minimize vendor effects, which I think is quite neat. I want to also give some practical considerations because there are so many sequences out there. It will be a bit generic, but I still thought I'd compile a little list of things to look out for and optimize before you scan 100 people on a particular protocol. The first one is calibration of RF pulses. We want to get obviously as much signal as we can. We learned that press in particular will perform poorly if you over or underpower the pulses. That's where pulse calibration comes in. And some systems do it automatically, but others need a little bit optimization. And this is something that's to be quite consistent for a given brain region. So it just pays off to pre-optimize that before running a study. The same is true for calibration of water suppression. As with sequences, there are many different ways of doing that, but most of them offer some sort of tweaking, interpulse delays, or flip angles or something like that. It's also quite consistent for any given brain region between subjects. Finally, a few important points to reduce unwanted signal from outside the voxel. So that particularly concerns spurious echoes or ghost signals. And one thing that can be modified is the order and the polarity of the size selective gradients. It's not automated, so that's something that you can pilot ahead of time before you start a study, just cycling them all through and checking which one gives the least strong artifact signals. There are also some useful MRSHub threads that describe how to do that. Then most sequences offer some form of outer volume suppression or saturation bands. They excite signals specifically in regions where you technically don't want any signal from, but then they crush that signal. And so I think finally the crusher gradients are also helping to reduce those signals. They can be tweaked on some systems. And there's also some computationally optimized schemes like the dot-cops one that are increasingly becoming available. All right, there was a quick sail through current state of the art a bit of single voxel. And conceptually, I always think of single voxel as a little bit inherently wasteful because you excite 3 entire slices and then you just throw most of the signal away. And there should be a way to be a little bit more efficient. You get some greater spatial coverage. And that's all what MRSI is about. And I thought I'd give a quick run through to just the signal generation concept, because we're hearing a lot about accelerated MRSI later on. So for those of you who are not necessarily that much exposed to MRSI, I thought I'd include a quick primer. So suppose you have 4 tubes at different spatial positions, and each of them has a different metabolite in them, right? And if we just acquire a spectrum of their entire thing, we'll get all the peaks in the spectrum there, but we don't know which peaks come from which tube, right? So we somehow need to find a way to encode the spatial origin of a signal into the signal. And in MRI in general, there are really 2 basic methods of doing that. And the first one is frequency encoding, where during signal readout, there's a gradient active. And let's say I'm in the magnet, and then the spins in my head have a different Larmor frequency than the spins in my feet. And that's how we can tell them apart. But that can't work with MRS, right? Because we need the frequency of signals to still encode for the difference between the metabolites, right? Our chemical shift, that's the information that we require. Fortunately, the other principle, phase encoding, that's still available to us, and it's pretty much available with all the sequences that we've seen before. And Robin de Graaf has a fantastic illustration explanation of that in his book. I thought I'd visualize it a little bit. So here we see the same experiment and the spectrum that it generates. There's no spatial information right now in that spectrum. We can't tell apart where the signals come from. But now we add a brief phase encoding gradient right there. And during that gradient, Larmor frequency again becomes a function of the position of the spatial coordinate, right? And after we switch off that gradient, each position along this tube has acquired a different phase, right? So we can do that again. We can increase the gradient just a little further. And then we can look at the signals that we want to trace. And if you look at that, the NAA peak has acquired just a little bit of phase over increasing gradients. If you look at the choline peak, it has acquired quite a bit more phase, but also in the opposite direction, right? And if we want, we can do that a few times actually, also with negative gradients. And we can take these signals and we can plot the integral of our signal over the strength of our gradient, right? So you see here that NAA does vary with a change in gradient strength, but it does so relatively slowly. The imaginary part is shifted to the right. And you see that choline varies faster with the imaginary part shifted left, right? Now, the main conceptual difference to realize between MRSI and single box spectroscopy is we're acquiring signal from the entire object all the time. We're not placing small repetitive voxels around each tube, right? So each time we scan, we get this complete spectrum here as a function of the gradient strength. And each time it is the sum of the all signals originating from all spatial positions. And each spatial position is modified by a characteristic phase factor that depends on the spatial position of each signal. So let's look at these signal trajectories a bit in detail, right? So you'll see that the phase, the signals of NAA and choline here, they change with a certain periodicity over the gradient strength. There's an oscillating pattern to it. And so the frequency with which that signal changes with gradient strength now encodes the position in space where that signal originates from, right? And you can see this frequency is relatively low for NAA, has a big wavelength, right? So the gradients at the position of that tube where NAA is in, they can't really be very strong. And it turns out that we can determine that this phase exactly corresponds to a position of x equals 1 centimeter. Now, the frequency is higher for choline, and the phase changed with opposing polarity. And that behavior corresponds to a position of x equals -2 centimeters. So one by one, we can really go through there and figure out which tube contains which metabolite. Obviously, we don't do that by hand, but we do that with a mathematical operation. And ultimately, again, at each gradient step here, we acquire the signal from all the positions from the entire object encoded by a phase factor. And that depends on only on the position of each signal. And we can replace the gradient area in this equation with a single coordinate k, which makes it a little easier. This is slightly handier notation. And by just swapping this around with the inverse Fourier transform, we've essentially done 1D MRSI already. So the spectra at each spatial position here is a function of the spatial coordinate r. Here, the Fourier transform of the phase encoded spectra depending on the gradient strength over here. And we can easily extend that concept to 2 and 3 dimensions. So we can do 2 and 3D MRSI by doing a 3D and 4D Fourier transform. Now that's great. So now instead of doing single box MRSI, we're throwing all of our signal away. We actually acquire signal from the entire object all the time, but that is not without cost or not without complications, I would say. Problem with phase encoding is that it is quite time consuming, right? We can only acquire one setting per TR, one gradient strength step per TR, right? And that becomes a little bit tricky if we try to think about spatial resolution of the outcome for a little bit. Just to quickly bring you up to speed about Fourier series, we can approximate pretty much any signal as a sum of periodic sine waves with a certain frequency. The problem is if we want to build really sharp transitions, we'll need a lot of components to do that. And what are sharp transitions? Well, ideally, if we do imaging, right, we want to have a sharply defined voxel where the signal really only comes from that voxel and from nowhere else. But anywhere in imaging, you have some sort of trade-off where that necessarily isn't always the case. So in the context of MRSI, now you can think of each gradient strength step that we do as offering us one component that we have to build a spatial resolution grid, so to say. But since each of these steps costs us so much in time, we can only do maybe, I don't know, 30 of these steps in a reasonable scan time. If we wanted to achieve a perfectly rectangular signal localization to a square voxel, we would need an infinite amount of these phase encoding steps, which is obviously not feasible. But instead, we have to settle for some sort of trade-off. The signal actually will come from all over the sample, as we just learned, which is kind of also the strength of MRSI. Oops. But it's now determined by a so-called point spread function. And of all of these signals that we acquired up here, they all count the same, they be weighted the same. This behavior will be described by a sinc function, right? So this means that there are side lobes to the point spread function that will fold in signal from the edges of the sample. And it can be mitigated to some degree, it should be, by weighting these different steps differently. And that's called so-called K-space apodization. So spectra acquired with a higher gradient strength in that setting will count less according to a kind of Gaussian or a Hamming filter distribution as shown here. And that helps downweight those sinc wiggles, but at the cost of a broadened central lobe. So you're kind of blurring out your nominal voxel in size a little bit. But it still helps quite substantially, especially now if you remember the first slide again, right? There are lipids all over outside the brain, and lipids give strong and broad and really unwanted signals that we want to get rid of in the first place. And even with K-space apodization, they can remain problematic. So sometimes you have to take extra steps. I know that I think Mikhail is going to tell us more about this in his talk later on. I wanted to tease it a little bit here. Briefly talked about outer volume suppression earlier. This is useful here too. So you pretty much excite signal where you expect lipids, and then you dephase them with a Crusher gradient, right? And as you can see, that can work really well as an additional measure to reduce these contributions. And that's pretty much it. So if we all wrap it up, single box spectroscopy offers us a nice way to be very laser focused on a particular region by throwing a lot of signal away, but offers high quality spectra in a reasonable amount of time. There's plenty of implementations available on scanners. They're even standardized, so they're getting increasingly standardized now, thanks to the efforts of many folks in the room here. Obviously, MRSI overcomes the greatest down point of single box spectroscopy, the lack of spatial coverage. Scan times tend to be a little bit longer. And as we will learn later on, as the acquisition gets more involved and more advanced, the post-processing and the analysis can get a little bit more involved. But we'll learn a lot about that in the dedicated sessions about that. Since we only had about 20 minutes, I had to gloss over a lot of the details of the, let's say, adiabatic pulses, or well, how does a stimulated echo form. I really highly recommend these resources for background reading. Robin de Graaf's book is an absolute Bible. It's fantastic, chapters 5 to 7. Almost all of the illustrations come from there. I really enjoyed Carl's review of the theoretical description of the single box sequences, and obviously the expert consensus on how to do proton MRIs nowadays is fantastic. And with that, I would like to acknowledge everybody I'm working with, near and far, and acknowledge my funders. And thanks. Have a great workshop. We're totally light.
Thank you very much for this nice review. Is there any question? Did you hear me before? I think it was not on. Did you hear? Okay. Is there any question? So
good.
I don't have any. I mean,
That's
there is
good.
plenty of questions, but I mean, for a basic.
I mean, it's
presentation,
supposed to be a primer, right?
Yeah,
so I'm glad there aren't that many questions coming up.
Okay, thank you very much.
Thank
so
you.
we will directly move, and this way we will stay on time. So the next presentations, the next talk will be given by Steve Williams from the University of Manchester, and he will explain to us and talk about the content of this spectrum or spectra that we can acquire with our MRI system. So okay, thank you. So Steve,
yeah,
it's your
thank you.
time.
Okay. Yep. Yeah, that's fine. Okay. Okay. That was a great talk, by the way, Georg. I mean, there's hours worth of questions on your talk, so I think we'll come to that later, but it's a good introduction. Yeah, so thanks to the organizers, particularly Christina, for asking me to give this talk. I think she asked me because she knows this is really tough to do in 20 minutes. So I said, I hope it's okay if I keep going till lunchtime, but she said not. But anyway, so this will try to get through this in 20 minutes. And I'm talking about the neurochemical profile. And I'm going to be talking about the detectability, the physiology, and the biochemistry of most of the major metabolites, which we can detect by in vivo proton MR spectroscopy. So I'd like to start with this slide, which is actually an extract prepared from a sample of human brain taken during the course of surgery. So it's actually normal appearing white matter, and the surgeon chopped it out and we dropped it in liquid nitrogen, and then we extracted it. So what you see here is at high field 11.7 Tesla, which when we did this was ultra high field. Now, of course, we can do human spectroscopy. Well, some of us can do human spectroscopy at 11.7 Tesla. At that time, this was a high field for chemistry. And really what I want to point out is there are a lot of signals we can detect in the human brain. And if you go down and look at the signals down in the baseline, you can see there's still a lot going on. So a lot of chemicals are detectable in the human brain by MR spectroscopy. So this is a list of 19 metabolites which we can detect. I'm going to talk briefly about all of these. There are some which you can see in human brain, which I'm not talking about. I'm not going to talk about oncometabolites, and I'm not going to talk about those signals which are on the other side of the water. So it's just these 19. And actually, a few months ago, we were discussing what you might want to include in a basis set if you want to analyze a human brain spectrum. And so actually with my colleague, Caroline and Laura in Manchester, and Paulina, who's actually in the audience, we decided to make up a solution containing all these 19 guys, but plus a lot of other chemicals, which are potentially in the human brain. So we made up a phantom containing about 30 metabolites, and we were going to run it on a clinical system. But before we did that, we also thought we would go to super ultra high field, so 18.8 Tesla, 800 megahertz. So that's definitely ultra high field. So this is the brain mimicking phantom. And again, rather like the extract spectrum, you can pretty much detect signals from all 30 of these metabolites if you're working at ultra high field and you've got 1 hertz line widths. It obviously gets more difficult if you go to lower field strength. Let's just give an example of why we're always pushing to higher field strengthening for MRS. And let's look at the creatine phosphocreatine region around 3 ppm. And you can see when we're at 800 megahertz, you can easily detect creatine and phosphocreatine independently. And you can also see that the GABA and the glutathione are shifted away from the creatine, shifted away from each other. That's great. We can measure them all. If only we could go to 800 megahertz and have 1 hertz line widths. If we run our phantom at 3 Tesla and we look at the same region, so this is a T35 millisecond press, and we look at that region around the base of the creatine peak, and you can see that now there's no possibility of separately analyzing creatine and phosphocreatine, and also GABA and glutathione have both moved under the creatine peak. They both overlap each other, and they're also sitting on top of aspartate. So we have to work a little bit harder in terms of both acquisition and analysis if we want to detect these relatively lower concentration coupled metabolites when we're working at 3 Tesla. So if we look at the typical 3 Tesla spectrum, dead easy to see the major singlet resonances, NAA, creatine, phosphocreatine, and choline. And then we can pick up a couple of resonances which are present at high concentration, inositol, glutamate, and glutamine. If you want to measure other things, we've got to have more advanced processing techniques and potentially different types of acquisition techniques. If you work at 7 Tesla, things look better. There's better separation of peaks. You can start to convince yourself that, yes, there is a glutathione right on the base of the creatine peak. And we can sort of start to tease out the GABA next to the NAA at about 1.9 ppm and at 2.3 ppm. But again, difficult to quantify those without being fairly sophisticated. Okay, so let's move from detectability to thinking about metabolites which we can detect. So I basically divide them into neurotransmitters, which I've called ISH, you'll see later, energy metabolites, antioxidants, membrane-linked metabolites, and then 2 more sort of functional definitions, markers and others, which don't fit any of these characters. So let's populate these. So the neurotransmitters, glutamate, GABA, aspartate, and glycine we're familiar with. I've included glutamine, which is most definitely not a neurotransmitter, but is intimately linked to GABA and glutamate metabolism and is involved in neurotransmitter recycling. So I've included it among the neurotransmitters to count as the ISH. Energy metabolites, the creatine, phosphocreatine, glucose and lactate all report on energy metabolism. The antioxidants, glutathione is the one most commonly measured, but as you'll also see, and has been done just down the corridor from here, as also ascorbate. Then the lipid metabolites, so glycerophosphorylcholine, phosphorylcholine, and also to an extent phosphoethanolamine. In the markers categories, I've put NAA and myoinositol, and I think most of you will know why they are there. And then the others, the related compound to NAAG, sialoinositol, and taurine. So I'll be talking briefly about all these. So let's first of all think about the neurotransmitters. So these are chemical messengers which transfer signals across the synapse from one transmitter neuron to one receiving neuron. 3 major classes: the amino acids, so the glutamate, glutamine, glycine, and GABA, present at high concentration, millimolar-ish. The monoamines, such as serotonin, dopamine, etc., they're down much lower concentration in the micromolar range. And then there's neuropeptides, things like vasopressin, oxytocin, much lower concentration. So the only things we can access by MRS are the ones at the millimolar concentration, so glutamate, GABA, aspartate, and glycine are all in principle detectable by 3T using MRS. And glutamate and GABA are fantastically important. They're the major excitatory and inhibitory neurotransmitters in the brain. They're involved in pathology, glutamate particularly through excitotoxicity. A lot of studies in psychiatric diseases showing imbalances in glutamate and GABA between disease groups and control groups. No real consensus on what this always means, because a lot of these psychiatric studies are confounded by medication, age, gender, etc., etc. But glutamate present at high concentration, GABA present at a detectable concentration. Glutamate and glutamine, we still benefit from going to higher field if we can. If you look at the spectrum on the left, on your left, the 3 Tesla spectrum, and I've the shaded box shows the glutamate C4 resonance, and you can see it completely overlaps or largely overlaps the glutamine C3 resonance, glutamine C4 resonance. And so this is why people quite often refer to GLX, the sum of glutamate and glutamine, because you can measure that with a greater degree of certainty. If you move to 7 Tesla, however, on your right-hand side, you can see you can get complete separation of glutamate and glutamine. So another benefit of going to the higher field strength. I want to say a little bit about glutamate's metabolism. And this links into why it's important to also measure glutamine. So if we look on this side, so glutamate in the center is in biochemical equilibrium with the tricarboxylic acid metabolite alpha-ketoglutarate. Glutamate can either be formed by glutamate dehydrogenase, which adds an ammonium directly to alpha-ketoglutarate, or it can have the ammonium moiety transferred from either aspartate on the left or alanine on the right. So a number of ways in which glutamate can be made, but it's always in equilibrium with the tricarboxylic acid cycle immediate alpha-ketoglutarate. When glutamate is released into the synapse, it gets taken up by astrocytes, and there it's converted to glutamine by the enzyme glutamine synthetase, which basically adds an ammonium to glutamate. And this enzyme is only present in glial cells. So this reaction has to take place in glial cells. The glutamine can then be released from the astrocyte back into the extracellular space and then get taken up by the neuron, where it is reconverted to glutamate by an enzyme which is at high concentration in the mitochondria of neurons, and that's phosphate activated glutaminase. And that actually pinches the ammonium off glutamine, converts you back to glutamate. So just an aside, every time you recycle a neurotransmitter from glutamate back to glutamine, you also pump ammonium from astrocytes into neuronal cells. So the neurons have to deal with that in some way, and that's something which we don't usually consider. GABA measurement, I would contend it's not detectable under normal conditions at 3 Tesla. I'm not convinced you can do it at 7 Tesla, although other people will. The next talk by Kimberly Chan is going to be describing spectral editing, so I'm not going to spend any time on this, but to say we need a way of suppressing the unwanted signals and detecting the signal we want. So we have on this slide an example of a megapress editing spectrum from GABA, where you can see the GABA signal is picked out in the middle. We also edit glutamate and glutamine. I was hoping for a more recent reference to this. So I know Georg has put together a fantastic article on how to measure GABA, beginner's guide, but I don't think it's out yet, is it? Okay. Yeah, okay. But anyway, if not, you can go back to the one which came from the GABA workshop way back in 2013, 2014, which tells you how to measure GABA. Aspartate, as a neurotransmitter, aspartate has attracted much less interest than glutamate, but nevertheless, it does classify as an excitatory neurotransmitter. Is it detectable by MRS? It's certainly detectable. So on the left-hand side, we have a high field short echo semi-laser at 9.4 Tesla, which some people in the audience, at least one I know, was involved in. So there is aspartate there. So the aspartate protons just to the right of the NAA around about 2.8 ppm. You can see they're blown up there. So yeah, definitely detectable, but how well you can quantify it, I think, is and what sort of effect size you would need to measure differences is another point. You can also do it by another editing technique, which I guess Kimberly may. Oh, sorry, need to go back. By another editing technique, which maybe Kimberly will talk about. This is Hermes. So I'm showing here editing, co-editing in a single acquisition the aspartate protons of NAA, NAAG, and aspartate. And yes, again, you can see aspartate, but I've been much more confident measuring NAA or NAAG by this technique rather than aspartate. Okay, I'm going backwards, something like that. I'll go forwards. Okay, so glycine, it's a neuromodulator, particularly NMDA glutamate receptor, so a pretty important compound, particularly in plasticity, learning, and memory. It's also elevated in brain tumors, well, I'm not really talking about oncometabolites much. Difficult to separate from myonosol because it has a single signal sitting at 3.55 ppm. Inositol is sitting right on top of it. You can, a couple of ways you can do it. You can go to long TE, use multiple refocusing. So basically you modulate away the inositol signal and you reveal the glycine poking out from the middle. You can also do that short echo, particularly if you go to high field. And again, it's a matter of playing around with your echo time to try and modulate away the myoinositol, so you're left with the glycine signal. So those are both possible. Energy metabolism, not really going to show you any examples here. Just point out glucose. Yes, we can measure glucose by proton MRS. It has been particularly useful for those people who are interested in modeling carbon-13 metabolism, because from proton MRS, you can measure the brain glucose concentration in situ in vivo. Lactate is not generally detectable. It's sitting at 1.33 ppm, right underneath the macromolecule lipid signal. So you can't really see it at 3 Tesla or I think at 7 Tesla, unless you do spectral editing. If you do spectral editing, then you can pick up lactate very easily because it's about 1 to 1.5 millimolar concentration. But if it's elevated, for example, in brain tumors or hypoxia, anoxia, then you can also detect it without editing. Creatine and phosphocreatine, I'm not going to do anything more than say these couple of sentences on them. Vitally important for energy metabolism because phosphocreatine provides a buffer for cellular ATP. As we can't distinguish creatine from phosphocreatine, we basically see the sum of them. And there was a tendency, certainly up until the mid 2010s, to say, well, we can use creatine as a concentration marker. And actually, there are quite a lot of diseases in which creatine concentration changes. So if you have a disease group and you want to compare it to a control group, not a good idea to use creatine as your reference standard. Markers of compartmentation, so I'll spend a couple of minutes on this. NAA is, I think everyone's familiar with this as a neuronal marker. Myoinositol is very often referred to as a glial marker. So yes, NAA is expressed in neurons, although actually it's also expressed in some glial cells. So this spectrum is something we did back in the 90s, where we did cell cultures of neuronal cells, made extracts and run them at high resolution. So you can see a very clear NAA signal in these cerebellar granular neurons. If you prepare cortical astrocytes, there's literally nothing there where the NAA should be. We've also analyzed these by HPLC, which has much higher sensitivity, and yet there is no NAA in astrocytes. However, we did see it in a oligodendrocyte precursor cell, the O2A progenitor, that has a clear N-acetyl aspartate. And one of our co-authors on this paper, the Oraniak et al., Kish Baku, when he was working in Oxford, he also showed you could detect it in immature oligodendrocytes. So yeah, NAA is in other cell types in the brain, but predominantly it is neuronal. What's it doing? Is it just telling us there are neurons there or not? Well, no, it seems to be a marker of neuronal function. So on this slide, this is work done in the 1990s at St. George's Hospital, where they had stroke patients, and they followed them for up to 100 days after stroke. And most of them were showing reduced NAA immediately after the stroke. But a couple of patients who ended up with good health outcomes, so they pretty much recovered from their stroke. They're the two ones highlighted in red. Their NAA recovered from a nadir. So NAA came back. So it's not that once you've lost NAA, you've lost neurons. You can actually just basically stun the neurons into being non-functional, but they can potentially recover and regenerate the N-acetyl aspartate. And a similar study has also been reported in rats, and reference is given on the slide. What is NAA for? Well, we still don't really know. Despite over 100 years of neurochemistry, we know it's synthesized in mitochondria. It turns over in about 24 hours. We know defects in the enzyme which breaks down NAA gives rise to very serious white matter disease, which is Canavan's disease. As I mentioned, it is present in oligodendrocytes. This sort of white matter related factor does suggest it may have something to do with acetate transport for lipid synthesis. And also it appears to be a reporter of neuronal function. So just briefly to talk about metabolism. So NAA is not usually broken down in neurons. It usually has to exit the cell, but actually it can be broken down by enzymes which are present on the surface of astrocytes into aspartate and acetate, or it can also be taken up by the oligodendrocytes, where it can release acetyl groups for membrane synthesis. Myoinositol, so I'm going to have to do this quickly because I'm on my 20 minutes. So this paper by Brand et al., very highly cited. The first one to show myoinositol is present in cortical astrocytes, it's present in glioma cells, but it's not present in neuronal preparations. And this is the basis of the contention that it's a glial marker. So that just shows the inositol. From our spectra, from neurons, actually these do contain myoinositol. And unfortunately, the only way I could see it was by scanning the spectra, because we no longer have access to these spectra. But yes, there is myoinositol in some neuronal preparations. So it may not be completely true to say that inositol is purely a glial marker. So I think I just need to leave this slide up. Well, the other thing I want to say about myoinositol is, interestingly enough, inositol, both myo and siloinositol, do stabilize beta amyloid. But I think it's unrelated to this is the fact that inositol also is elevated in Alzheimer's disease. I'm not sure if this is the first reference, but it's certainly one of the early ones to the detection of increased myoinositol associated with Alzheimer's. But that's now been reported many times, both in human cases, also in animal models, and in transgenic models. So just to summarize, the blanket assumption that inositol is a glial marker maybe is not true in all cases. The choline-containing compounds, I'm going to have to just whiz through this, but glycophosphorylcholine, phosphorylcholine are the major metabolites. Phosphorethanolamine is about the same concentration as GPC, but because it's only 2 protons compared to 9, it gives a much lower signal. So this again is one of these high-resolution spectra recorded from extracts. So in vivo, we can't separate GPC and PC. I said I wasn't going to talk about onco, so this is my only cancer slide. The interesting thing is that in normal brain, the GPC is greater than PC, but in tumor cells and tumors in vivo, we think the increase in choline-containing compounds is driven by an increase specifically in phosphocholine. Glutathione is the major antioxidant. It's a tripeptide, lots of signals, all overlapping other stuff. So our contention is you really need to measure it by spectroscopic editing. So you can see, you want to measure the glutathione by LC model fitting. Good luck with that. A lot of people reported at 3 Tesla, a lot of papers on it. My feeling is you really do need to edit it, where at least you can get a signal which you know is glutathione. Ascorbate, I have to include this slide because it's just down the corridor from here. Ascorbate is an important antioxidant, massively tough to measure in humans, if at all possible. You can do it if you have a nice high field scanner and you have someone like Christina to shim the magnet, or don't know whether you shim the magnet these days. You probably have people to do that for you. So actually, yes, so the ascorbate is sitting there with the other alpha amino acids, and you can, from fitting the spectra, measure changes in ascorbate, in this case with a disease, hepatic encephalopathy. Taurine, don't be fooled by the bull, that's not a cow. Taurine is from Taurus. Interestingly enough, it seems to mirror NAA in terms of what goes on in development. So taurine starts high and goes low during development. NAA starts low and goes high during development. Taurine's also big in the wellness industry. You get 1 gram of taurine from 1 can of Red Bull. if you drank 70 cans of Red Bull, that would be all the taurine you have in your body, about 70 grams. What's on the other side of the water? So I've only talked about things between 0 and 4.7 ppm. So on this side, on the side of the water, we're in now a lot of interesting stuff going on. This is a map of Europe from AD 830. So lots of interesting groups of people. You might wonder what they're doing these days. What's on the other side of the water? Well, something smaller, just Anglo-Saxons and Picts at that time. Now we've got Brexit, there's even more Anglo-Saxons and Picts and fewer of any others. Okay, there are peaks on that side of the water. I'm not going to tell you anything about them. So finally, acknowledgements. So I mean, this is based on a talk I wrote about 20 years ago. So I've been able to update it, particularly with the help of Lynn Uray's review. But I also like to acknowledge the people I currently work with in Manchester and in Oxford, and the people who work on JMIUI. So thank you for your attention.
Steve, thank you very much. Thank you for this very nice review. Is there any question? Sorry, I start with Obidu, who comes from later. Yes, sure.
I wanted to ask you about the how translatable are these? You actually alluded during the presentation the results obtained from cultured cells to humans. I know that especially in disease, model disease, that's very little translatable, or like partially, especially like this compartmentalization of myonositol glials and so on. Is it like you think once you take a specific cell type and put it into a culture, does it recapitulate everything that's in the human brain and so on? And like, is that the barrier? And the second question is, you mentioned something about GABA you think is not measurable even at 7 Tesla. Is it because of very low concentration, overlap, other things?
Yeah.
Thank you.
Okay. So in terms of the first question, no, you can't be sure. The cell culture does not recapitulate what you get in vivo. So we have some metabolites which we detect in the cell culture which aren't present in vivo. For example, taurine, which is very high concentration in rat brain, not as high concentration in human brain. If we culture neurons, they don't have taurine, they have hypotaurine. So it's not a one-to-one correspondence. So you have to interpret the cell culture data with some caution. In terms of GABA, it's a low concentration and the overlap. So the three signals from GABA, one is sitting basically underneath the base of the NAA. The next one is right next to glutamate. And the one we edit is sitting underneath the creatine. So I mean, you know it's there. And I think at 7 Tesla, people would claim if you do a model fit, you can measure GABA. I don't think that's the case at 3 Tesla. I think you really do need spectral editing at 3 Tesla. But it's due to overlap and low concentration. Oh, yeah, no, definitely. No.
I know that you everybody recommends spectral editing, but I thought you like when you mentioned it, you expressed doubt that even with spectral editing, you doubt that the GABA is measurable at 3 or even 7 Tesla. Or if you can clarify that, maybe I misunderstood.
Yeah, well, if I said that, I was definitely mistaken. No, absolutely, you can measure GABA with spectral editing. I hope I didn't. I don't know. I either misspoke or you misunderstood.
Thank you. Perhaps the last question, Christina.
Thank you very much. Choline compounds, can we consider them as glial metabolites or not so much?
No, they're definitely. Well, I think I think the choline concentration is higher in glial cells than neurons, and I think if you get gliosis, you will get an increase in choline-containing compounds, but they're certainly not specific. They're definitely in neurons.
Okay,
Okay,
with this
thank you.
last question, where I think we'll stop here. We'll have time to meet the expert later on or on the table.
Yeah, sorry.
So thank you again. And I will invite the next speaker. Yeah. Kimberly Chen will speak about editing in vivo.
Yeah, go ahead. Right, can everyone hear me? All right, cool. All right, so I'm going to talk about spectral editing. So first of all, what is spectral editing? So as we know, the proton MRS spectrum suffers from a lack of spectral dispersion. Because of that, there are many different overlapping peaks that can complicate quantification. This is especially an issue for low concentration metabolites, such as lactate, 2-HG, GSH, spartate, and GABA. GABA is actually shown here in the spectrum right here in green. As we can see, the spectrum is magnitudes lower than the proton MRS spectrum, and can be hard to detect conventionally, as we can see here, because of this large creatine peak over at 3.0 ppm. So quite broadly, spectral editing can be defined as any sort of technique that allows you to simplify the spectrum to limit detection to select metabolites. And there's many different types of spectral editing techniques, such as COSY, constant time press, J difference editing, but in the interest of time, I'm only going to talk about J difference editing, which is the most commonly used method. So the J difference editing is typically done using a megapress pulse sequence, which was first published by Mesher and al. In 1998, and consists of a basic press sequence with a 90-degree excitation pulse followed by 2 slice selective refocusing pulses. And in megapress, editing pulses are added about the second refocusing pulse. In the typical megapress experiments, we have 2 types of subacquisitions: an edit on acquisition, where these editing pulses are applied on resonance for your metabolite of interest, and the edit off acquisition, where these editing pulses are applied off resonance for your metabolite of interest. So when you subtract the 2 subacquisitions from one another, you remove any large or overlying signals that might be overlapping your metabolite of interest, and you get a reduced spectrum. So how does the spectrum respond to these different editing pulses? So I'm going to use GABA here as an example. So right here shows the simulated GABA spectrum in the off case. There are multiple resonances. For editing, the resonance of interest are at 1.9 ppm and 3.0 ppm. As we can see here, the 3.0 ppm spins, or sorry, 3.0 ppm resonance has inverted outer peaks. As it turns out, the 1.9 ppm resonance is coupled to the 3.0 ppm resonance. Therefore, in the edit on case, when we apply our editing pulse at 2.0 ppm, coupling is refocused, and we see a spectrum that looks very much like this. We see at 3.0 ppm a sort of triplet-like structure. And when we subtract the 2 from one another, we get a spectrum that looks very much like this. You typically detect the 3.0 ppm resonance for GABA, and you get positive outer peaks. So how does this look in vivo? So in the edit off case, you see a spectrum that looks very much like this, that looked like the spectrum that I showed you earlier. However, in the edit on case, when we apply an editing pulse at 1.9 ppm, we see the spectrum starts to change, most notably at 3.0 ppm, where GABA is located at. And subtracting 2 from one another, we see this reduced spectrum that looks very much like this, with a clear GABA peak at 3.0 ppm. Now, although most conventional spectral editing acquisitions are designed around targeting certain metabolites, truth of the matter is you don't get just that metabolite. You get a bunch of others. So let's say for GABA, as I shown you I showed you earlier, here's the spectrum, edited spectrum for that. And we see a GABA peak at 3.0 ppm, but we actually also get a bunch of other metabolites. So we also get glutamate and glutamine, NA and NAG, and also, annoyingly, macromolecules that overlap with the GABA peak. And this results from the fact that our editing pulse is not sufficiently narrow enough to just target GABA. You often affect other resonances as well that are within the bandwidth of your editing pulse. And sometimes this could be beneficial or detrimental depending on the frequency of the co-edited metabolite. So in the case of GABA, you get glutamate and glutamine. It might be interesting to measure glutamate and glutamine from this edited peak at 3.75 ppm. But for conventional GABA spectral editing, there is a macromolecule peak that you co-edit that overlaps directly with your GABA peak at 3.0 ppm. Let's also take GSH, for example. You see a clear GSH peak at 2.95 ppm, but there's also other metabolites too. So you get NA and NAG, and also lactate and macromolecules. And although lactate and macromolecules don't affect detection of GSH because it's far away from the GSH peak at 2.95 ppm, the NA and NAG peak are right adjacent to the GSH peak, which can complicate quantification. So one way you could possibly get rid of co-edited resonances that are unwanted is through symmetrical editing. And this is typically applied to GABA and separating it from macromolecules. So macromolecules suppress GABA. So in the conventional GABA edited experiment, you have an editing pulse that is applied at 1.9 ppm. And as we can see here, the editing pulse bandwidth also covers the macromolecule peak at 1.72 ppm. In macromolecule suppressed GABA editing, however, you reduce the bandwidth of these editing pulses so that you co-edit macromolecules to a lesser degree. And you co-opt this edit off to instead be at 1.5 ppm so that it is symmetrical around the macromolecule peak of 1.72 ppm. Therefore, in the edit on acquisition and the edit off acquisition, the macromolecules are co-edited to the same degree. And so when you subtract the two from one another, macromolecules are subtracted out. And so now I'm going to talk about a little bit about some practical aspects and some kind of like the current state of the field. So in spectral editing, you typically sacrifice SNR for kind of specificity. So in the typical spectrum, you get a whole bunch of different metabolites that you can measure. You can get glutamate, glutamine, creatine, phosphocreatine, NAA. However, when you perform spectral editing, you limit the spectrum to sort of a certain frequency ranges. And because of that, and also in addition to this, we usually measure low concentration metabolites with editing. And because of this, we normally use really long acquisitions, like 12 minutes, in order to get sufficient SNR. So people have started performing multi-metabolite acquisitions, so kind of multiplexing your experiment to acquire multiple metabolites within the same spectral editing acquisition. And there are several ways of doing that. The most simplest way is through optimized co-editing. So let's say that you normally get some lactate with GSH, but you're targeting GSH. With optimized co-editing, you can optimize the pulse bandwidth and also the frequency so that you get both lactate and GSH optimally. And then also I'm going to talk about DO and Hermes to detect multiple metabolites. So DO allows you to detect two metabolites simultaneously within the same edited spectrum. And this works as long as your detected signals are frequency resolved from one another. So in the typical megapress experiment, you have an edit on for your metabolite and an edit off for your metabolite of interest. In DO, however, this edit off acquisition is instead co-opted to be edit on for a separate metabolite. So each sub-acquisition kind of doubles as an edit on and an edit off for two different metabolites simultaneously. And this was first demonstrated to detect both ascorbate and GSH to form a antioxidant profile. And as you can see here on the right, bottom right, we see a due spectrum with both ascorbate and GSH, and but with opposite polarities from one another. One downside to this technique, however, is that your detected metabolites or any sort of co-edited metabolites also have to have resonance that are separated from one another. So let's say you want to do editing of GSH and lactate. You normally would apply an editing pulse of 4.56 ppm to target GSH, and an editing pulse at 4.1 ppm to target lactate. Unfortunately, this 4.1 ppm editing pulse also targets creatine as well, so which has couple spins at 3.9 ppm, but also at 3.0 ppm. So in your due edited spectrum, you get GSH and lactate as you desired, but unfortunately you also get a negative creatine peak here, which overlaps with GSH. Because of that, and there are often times where you want to detect metabolites that are overlapping each other. For example, like GABA and GSH, they both have detected resonances around 3.0 ppm. And normally with spectral editing, you wouldn't be able to, would only be able to detect one or the other. And in order to detect both of them simultaneously, you can use this technique called Hermes. So let's say you want to detect GABA only. So let's say you would need at least 2 ons and 2 offs, and this can be acquired in any particular order. And let's say you want to do GSH only. So once again, you need at least 2 ons and 2 offs, and this can be acquired in any particular order. As it turns out, you can detect both GABA and GSH simultaneously by acquiring all the ons and all the offs for both GABA and GSH, but in a specific order, A, B, C, and D here, so that the coding pattern for GABA and GSH are orthogonal to each other, sort of like the columns of the Hadamard matrix. And depending on how you add and subtract these different subacquisitions, you can either get GABA only or a GSH only spectrum. So let's say you want to get GABA in this sort of experiment. So you would take subacquisition A minus B plus C and minus D, and you get a GABA spectrum here because we're subtracting the offs of GABA from the ons of GABA. But simultaneously, you're also subtracting the ons of GSH from each other and the offs of GSH from each other. So the signal for GSH is essentially nulled out. From the same experiment, you can take subacquisition A plus B minus C minus D, and you get a GSH spectrum that looks very much like this because you're subtracting the offs of GSH from each other, and you're subtracting the offs of GSH from the ons of GSH, but simultaneously you're subtracting the ons of GABA from each other and the offs of GABA from each other, which nulls out the GABA signal. And in vivo, we see here that the megapress acquisitions of GABA and GSH separately is comparable to that of Hermes across all subjects and with the same SNR. So one annoying problem with J-difference editing is that it can be quite sensitive to frequency offsets, and this can come from various sources, most notably subject motion, or gradient-induced heating or cooling, which can result from like prior fMRI or DTI experiments, which run the gradients pretty hard. Or this can come from poor B0 shimming in the brain. Let's say you have your voxel is in a location that's near an air tissue interface, or you have a very large volume to shim over, let's say for like MRSI. And this can cause all sorts of problems. For example, you can get lower editing efficiency that essentially reduces the SNR of your spectrum and also leads to misestimation of the metabolite levels. You can also get subtraction artifacts from the subacquisitions being misaligned from each other. So let's say for GABA, when the ons and offs are misaligned from one another, you get a subtraction artifact from creatine at 3.0 ppm, and that overlaps with your GABA at 3.0 ppm. And in the case of macromolecule suppressed GABA editing, you can also get macromolecule contamination. As you can see here on the bottom right, macromolecule suppressed GABA is more sensitive to frequency offsets than macromolecule unsuppressed GABA. So how can we account for this and address this? So one way is through interleaved prospective frequency correction. So in this, you acquire water references instead of you acquiring them right at the very beginning. You intersperse them throughout the scan so that you can determine the frequency offset and update the frequency periodically throughout the scan. On the right, we can see that without this interleaved prospective frequency correction, the scanner drifts quite a bit over the total acquisition time. However, when you use this interleaved prospective frequency correction, you see that this drift is reduced significantly. However, you do still get significant B0 drift over heavy scanner use after heavy scanner use. So recently we came up with a way to kind of address this. So with MC-Mega, and MC-Mega allows you to do shot-by-shot prospective frequency correction. So in the typical megapress experiment, you have a water suppression module that precedes your megapress experiment. In MC-Mega, however, we replace this water suppression module with metabolite cycling instead. And this allows you to retain the full water and metabolite signals at each transient. And because you're retaining the full water at each transient, you can perform prospective frequency correction at each transient instead of, let's say, like every 20 averages. So in the top right shows a phantom scan after 33 minutes of diffusion MRI. And as we can see here, WS-Mega, which is the interleaved prospective frequency correction technique I described earlier, shows a sawtooth-like drift, while MC-Mega displays a more stable frequency over time. And MC-Mega also produces a nice GABA spectrum, such as the one shown here. So we found that MC-Mega led to a 40% lower choline artifacts than WS-Mega. Choline artifacts are already also 75% lower with MC-Mega. GABA concentrations in medial prefrontal cortex were also 43% lower with MC-Mega relative to WS-Mega due to lower subtraction artifacts and also macromolecules. And we found that while in the occipital lobe that WS-Mega is correlated with the frequency offset, this goes away with MC-Mega. So now I'm going to talk a little bit about retrospective spectral alignment. So although you can't correct for editing efficiency or macromolecule contamination retrospectively, you can account for line width and subtraction artifacts post-processing. And there are several ways to do that. Most simply, you can use peak frequency and phase alignment, and this can be done to the biggest peaks in the spectrum, so like water, creatine, and NAA. Or there's also spectral registration, which allows you to align the FID in the time domain, and also robust spectral registration, which was optimized for multi-metabolite editing. So there was a recent study kind of studying all these different retrospective spectral alignments, and they found that the creatine alignment produces the fewest outliers with the narrowest GABA line width and choline subtraction artifacts. Though this likely also depends on the type of edited acquisition you have. So it can be quite time consuming to collect all these different single voxel data from multiple regions, especially with this long duration around 12 minutes. So people have also started combining spectral editing with multi-voxel techniques in order to get fuller brain coverage. So one notable experiment was done with a spin echo pulse sequence with MRSI, sorry, slice-based GABA MRSI with a spin echo pulse sequence and dual band water and lipid suppression pulses. They got nice GABA spectra here, shown here, and a nice GABA map. And they found that GABA was positively correlated with gray matter, which is considered consistent with prior literature. And this was also taken a step further for GABA MRSI at 70. So here they used a 1D semi-laser sequence, adiabatic editing pulses in order to account for the greater B1 inhomogeneity present at 70. They also used real-time motion correction with 3D EPI navigators and concentric circle echoplanar trajectories. They also inserted here this worst inversion recovery, which was used here to perform macromolecule suppression in place of the MM suppressed one that I showed you earlier, because there's greater B naught inhomogeneity at 70. And they found here that they had nice spectra. They also found 2.4 times higher GABA in gray matter than white matter, again, consistent with prior literature. And they also got some pretty nice GABA maps from all 5 subjects that they studied. So perspective, so there, as I told you earlier, it can be quite detrimental to have motion in your scans, and this becomes worse in MRSI scans. However, you can't perform any sort of retrospective correction for MRSI the same way that you would do for single voxel scans, since each data point is acquired in case space and is thus phase encoded. So we recently came up with a method to allow you to perform retrospective motion compensation for edited MRSI. So what it does is it goes through each point in case space and it looks at the water peak for all sub-acquisitions. So let's say on 1, on 2, off 1, and off 2. So all the water peaks should in theory have all the same phase if there's no motion. However, if you do have motion, you would see a different phase, such as the one here for on 2, which for on 2. And so when you take the correlation coefficient between all these different water peaks, you get a lower correlation coefficient for the motion affected averages. So we use that to identify which ones were motion affected. We attempt to phase correct that motion affected average. And if the correlation coefficient is still less than 0.9, then we replace it with a sub-acquisition, similar type sub-acquisition that is unaffected by motion. And if correlation coefficient is still less than 0.6, we zero out the entire point in case space. So as we can see here, here's some represented spectra from our edited MRSI acquisitions. So in the uncorrected version, you get negative choline and creatine peaks that dominate the spectrum. However, when you perform this retrospective motion compensation, you see a clear GABA peak at 3.0 ppm. We also found that choline artifacts were 3.6 times higher in the uncorrected than the corrected spectrum. And we found that with motion compensation, we got smoother GABA plus maps than in the uncorrected version. So I'd like to thank my collaborators, especially Richard Eden, who provided me with a lot of these slides, and also our current and former grant sponsors. Thank you.
Okay, thank you very much. Showing these different ways to track the GABA in the brain. Is there any question? No? Oh, yes. Quick one.
Thank you very much for this nice presentation. Often we approach at 3 Tesla to measure both neurotransmitters, GABA and glutamate, and preferred ways editing. Can you comment maybe on the reliability of glutamate measurements in editing sequences? Thank you.
Yeah, so there's some recent work done that has showed that this edited glutamine signal is not correlated with the non-edited version. So it's likely that it might be more reliable with the non-edited version than the edited version.
Okay, yeah, just a quick question.
Yeah, it's a very quick question. I just heard that the creatine signal at 3 and 3.9 ppm are linked. I never heard of that before, so
Yes.
wondering.
There's a very small coupling, 0.3 hertz between the two.
Okay.
Yeah.
All right, thanks.
Okay, thank you again. So we'll move on. For the next part of the presentation, I will give the microphone to my colleague who will introduce the next speakers.
Our next speaker is Candice Fleischer from Emory University, and she will be giving us an update on MRSHub. Really quick, there's a microphone in the center aisle that you guys in the back can use to ask questions. Thank you.
Either way is fine. Fine.
Thank you. All right, thank you so much. Thank you to the organizers for this wonderful workshop and to the moderators of this session. So I'm here today to give an update of the MRSHub on behalf of the Code and Data Sharing Committee. And the committee for this year, Kelly Swanberg and I are staying on for another year. And then we welcome Antonia Kaiser, Jamie Neer, John Lemaster, and Helga Zollner. And the purpose of the committee really is, this again, Christina mentioned this was started in 2019, but this is a standing committee within the study group. And really the goal is to really promote open science. So open education, data sharing, and really creating tools that facilitate this exchange, really not just within the ISMRM, but around the world, and really focusing on all users of MRS. So everything that we do in the committee and really towards this initiative is hosted on the MRSHub. And again, if you're not familiar, I just want to go through what really is in the hub and to promote people and really encourage people to use this. So on the homepage, this is really just a call for any sort of activities that we're doing or announcements such as this workshop. And we're going to be asking a lot from the community over this next year. We have a couple of projects, which I'll talk about, and those will be posted here. So all of the announcements will be on the homepage. Our major project, like Christina mentioned last year, was creating an educational resource. And we really want to promote this early in the workshop. So for those of you who are new to MR spectroscopy, you can go to this resource and look up any questions that you might have. So we created this community-sourced ebook, which has not just slides, but also curated materials and a glossary, which was some of the big contributions. And again, this was the work of the committee last year. So again, if you're new to MRS, we encourage you to check this out. This glossary was really designed for newcomers to the field. There's a lot of really excellent consensus resources that exist. But again, when you're first starting, many of our trainees and our students have no introduction to MRS other than maybe a chemistry course. And so this was really written with those trainees in mind. So the hub also hosts a lot of software and code sharing, and we really encourage you to use this. We're trying not to reinvent the wheel every time we need to do pre-processing and analysis. We also encourage you to post code and software that you're willing to share here on the hub. The forum, like was discussed earlier, this is a really great place, and I'm really going to encourage everyone to use this during the meeting and beyond. But this is for everything from questions. If you're not sure how to do something, again, you're just starting out, or you're interested in collaborating, for example, this is a really great place that we can connect with the community. And one of the things we really want to promote is, you know, an ISMRM in many ways is a society closed to ISMRM members, which I guess is by design. But MR spectroscopy is much larger than just the ISMRM. And we really want this to be a place for people all over the world, particularly those in limited resource countries or places where, you know, the ISMRM isn't as present, to really be able to network and discuss science in the field of MR spectroscopy. And then lastly, there's data sharing. So if you have resources that you want to share, or you need a data set for some validation, which I'll talk more about in a little bit as well, but this is a really great place to share data and to access data if you need something to try out a new method, for example. All right, so our projects for this upcoming 2022-23 year, we have three main goals. So the first is a continuation of a project that Kelly Swanberg led last year as part of the committee. And it's this Proton MRS VERY, or Validation Effort Resource Initiative. And the VERY is going to, currently it's hosted on GitHub, but the idea is that it will contain phantom data, synthetic data, and then MRS and non-MRS data combined. So this would be something like MRS data and PET combined in the same area, or MRS and ex vivo NMR. And I think her vision really was that this is very difficult data to acquire and obtain. And so wherever we can share that would be ideal. And so the effort for this year is to wrap this project up and to write a paper so this site can be posted and cited. The second thing, and what we really will be asking from the community, is developing a working group for synthetic data generation. So in the next few months, you should expect a survey from us on best practices for generating synthetic data. And what we really hope is to fill the gap in consensus on synthetic data. There was a number of papers that were published in consensus in the NMR Biomedicine edition last year, but synthetic data wasn't one of them. So what we're really hoping to work towards is consensus in this area. And we're really hoping that members of the community, many of you in this room, will be, you know, involved in this working group. This isn't just the committee. We really want everyone who wants to be involved to be active. And then the last thing, this is really Georgia's brainchild, is to try to develop an MRS hackathon for the first time. And what we're planning, or envisioning maybe is a better way to say it, what we're envisioning is before ISMRM Toronto next year, 1 to 2 days prior, we'll have a hackathon with a very clear goal in mind. And the goal will be to generate something that can be used widely. So we haven't finalized this. Again, you'll be expecting a survey from things that might be interesting to you. One of the things we discussed was maybe a canonical basis set generator, where you could, for example, input a header and generate a basis set in some format, for example. So if there's any other ideas you have, again, we would love to hear them. And if you're interested in helping or participating, we'd love to hear that too. All right, so I want to end with just kind of our vision for what we could have as a community and really what we could do with the MRSHub. So if you haven't gone into it, this is the QR code for the website. So for trainees, what we really want to encourage you to do is start here with your learning. Again, everything on this site was really designed for new users of MR spectroscopy. So use those educational resources that we compiled. We're really hoping that that educational document will be a living document, and I'll talk about that as well. Ask questions, so use that forum to your advantage. Ask questions as much as possible. And then as you're developing tools or data sets, we encourage you to post them on the hub as well. So for more active researchers, we encourage you to engage in the forum, answer the questions, develop new collaborations, new discussion topics. Hopefully you can start that at the workshop. Again, encourage your labs to share data and software and other resources that might be helpful. And if you're giving presentations such as these, which are really educationally focused, we encourage you to post those and share them on our educational resource. Again, we really want that to be active where people are regularly contributing. And then for leaders in the field, again, we want you to participate. We want you to involve and be engaged in the forum. And we also want you to share your resources. So even if you're not sharing data or being active, you can still share those educational presentations that you're giving at these types of workshops. All right, and again, for everybody, we want you to use the forum. I think we've had some challenges with the MRS study group email listserv. And so this is a site that's really separate from ISMRM that can really be used to form collaborations, ask questions, and have discussions. And spread the word, let people know, especially the trainees in your labs. I know my trainees have really benefited from these educational materials, so we really encourage you to use them. All right, with that, I'll thank you. I want to end with just a shameless plug that we're looking for students and postdocs in my lab. This is the beautiful city of Atlanta and our new imaging center, which houses 7T and a number of other MR tools. So with that, I'd like to open it up for any questions or discussions related to the committee or the MRSHub. Thank you.
So yes, I have just a question. Do you, in the data sharing, do you have room for helping diagnostics, for instance, comparing spectra for different disease or different conditions?
Yeah, I think that there is some room for some data sharing. I think, George, can you comment on that maybe briefly? Sorry to put you on the spot.
I think it's pretty much, well, we try not to host data ourselves, but it's kind of an aggregation of links. So we don't have terabytes of storage. I think what will emerge over time is Alex Lynn is leading an effort to do large-scale data aggregation of clinical data on his service. But I think, it's not something that we immediately do at the hub. It's rather for example data sets to demonstrate a certain method, right? That kind of
More educational. Okay. Yeah. Thank you. I know if there are other questions.
Hi, my name is Kasia Wasilewska, and today I will focus on the question, does visual stimulation always lead to an increase in glutamate concentration? Functional magnetic resonance spectroscopy measures neurometabolic concentration changes during the brain activation related to task performance. Action potential in the active brain area causes the release of glutamate to the synaptic cleft. Hypothetically, glutamate released to the synaptic cleft has a longer echo time, which could cause a higher signal in spectroscopy than glutamate stored in synaptic vesicles. Here you can see a theoretical plot of two spectra, the blue one from the rest and the red one from the activation. And we can expect a stronger glutamate response after the stimulation. Most of the previous ephemeris studies used very simple visual stimuli, like checkerboards as compared to a black screen, with the region of interest placed in visual cortex. Typically, functional magnetic resonance was conducted in block design, where the stimuli are presented for a long time. After averaging acquisitions from baseline and stimulation separately, we can observe two spectra with a slightly different glutamate response. Since we were interested in ephemeris changes related to reading skill, we have decided to use more complex stimuli than in previous studies. We have used two types of visual stimuli, words in Polish and false phone strings. Stimulation consists of 3 words presented for 250 milliseconds each, with a 15 milliseconds break. The target is a word with a letter or a false phone string with a descending feature like J, P, G. In our project, we acquired data from 3 regions of interest. 2 of them were chosen from a reading network. First one, the visual word form area, VWFA, which responds more strongly to a written word than other visual stimuli. And the second one, a left superior temporal sulcus, SVS, activated on visual and auditory stimulation. And medial prefrontal cortex as control region, not directly involved in reading process. Here we can see a theoretical glutamate response function created by Professor Mullins based on 4 different studies. We still don't know how exactly glutamate concentration changes in time after the stimulation. We tried to create an ephemeris paradigm that helped us to answer this question. We try to achieve that. To achieve that, we've decided to have 4 different time intervals between stimuli onset and data acquisition. 500 milliseconds, 1000 milliseconds, 3000 milliseconds, and 4500 milliseconds. Here we can see a graphic representation of our paradigm. These green bars represent visual stimulation. 3 words or 3 false phones. MRS signal is acquired with 4-second repetition time. The stimulation block contains 16 data acquisitions. It takes 64 seconds. And in each stimulation block, we have first signal of each type of time interval. In the experiment, we have 13 stimulation blocks with the rest between them lasting around 32 seconds. STS and VWFA are placed based on individual activation. The fMRI experiment on 3 Tesla MR scanner consists of 2 visual and 1 auditory, 1 big task. During the fMRS experiment, head position in the scanner is not the same as in fMRI experiment, which is why co-registration is required. And after that, we can take the coordinates of STS and VWFA and put them as input in the voxel position on the scanner. Scans were performed on Siemens 3 Tesla Trio system with a 32-channel radio frequency coil. Here we can see data acquisition parameters of our study. We examined 37 healthy right-handed subjects, 16 to 24 years old, with different levels of reading skills. MRS signals from words and false phone strings were analyzed separately. The FID-A MATLAB toolkit was used to preprocess MRS data, divided into 3 groups based on the time log between stimuli onset and MRS data acquisition. An absolute glutamate concentration was calculated using LC model. And within subject ANOVA with regions of interest and part of the spectrum, rest short long, were analyzed. We analyzed STS and medial prefrontal cortex together within ANOVA model. We did not include VWFA estimates due to a large amount of missing data. We can observe statistically significant differences in glutamate concentration between rest and short and between rest and long for both types of stimulation. We did not find an effect of the region of interest or an interaction between region of interest and measured part. As a comparison to glutamate response, here we can see concentration changes in total creatine. Only for a worse condition, significant change between the rest and the long and between a short and long can be observed. And, this observation could be caused by the Bolt effect. In conclusion, we obtained the main effect of post-stimulation intervals, and means spectrum parts split in terms of time log between stimuli onset and data acquisition. According to the almost equal concentration of total creatine in rest and short, we can conclude that glutamate increase is driven by metabolic shift rather than the Bolt effect. We've also shown that in our project, glutamate response does not depend on the type of stimulation and region of interest. In the future, we want to examine our subjects on a scanner with a higher magnetic induction and include this data to the analysis. Also, I hope that thanks to our paradigm design, we will be able to explore glutamate response function. We can split our data into 4 smaller groups. We need to analyze data from VWFA. So far, we have data from 15 subjects. But we believe that we will be able to achieve good quality data from this region using 7 Tesla scanner. Thank you very much for listening.
Are there any questions?
Thank you. Very nice presentation. Was I right that the voxel size is 1.5 by 1.5 by 1.5? That's 3.4 cc?
Yes, it's 15 by 15
Tesla?
by 15 millimeters. Yes.
Yeah. Can you maybe tell us a little bit about the Kramer-Rao lower bounds on the glutamate that you got from very small, such small volume at 3 Tesla that allows you to reach those conclusions regarding those statistically significant differences? Thank you.
Thank you. We had to use this small voxel because this region of interest, especially VWFA, is placed in very difficult position in the brain. And this is why we can't use a bigger voxel. We take care of this quality data, and right now it's hard for me to remember the exact boundary which we had there, but yeah, sure. Sorry, I'm very sorry that I didn't mention about that during the presentation.
Okay, I have a comment related to the assumption what glutamate is doing during or after excitation, because you presented a slide about some simulation about what is possible going on with change over 10% of glutamate within half a millisecond.
No, we had a 3% changes.
Well, still, it is based on my understanding of biochemistry and also what Steve beautifully showed in his talk today, that basically what we see is a glutamate as a metabolite linked to TCA cycle, and the amount what is going to the cleft of synaptic cleft is so small, so the change or influencing is for me basically marginal. So what we see during functional spectroscopy is changing into the function of a TCA cycle or the rate, not necessarily glutamate in the cleft. So yeah. I don't know if you can comment on it.
No, we're not sure if this is the right interpretation that this is caused by the glutamate release to the synaptic cleft. This is still preliminary results, and we consider a few options.
Yeah, okay.
Maybe this is a similar comment related to the glutamate, Boulder effect. You mentioned that the Boulder effect is visible. Now, line is getting narrower, means effective SNR is getting higher, especially like at 3 Tesla, glutamate quantification would be related to separating from other signals, and the glutamine is another thing. Yet, if SNR is higher, means maybe your apparent concentration may be higher. So we have actually posters showing that the SNR affecting the concentration quantification result. So that would be interesting to consider when you interpret your data in terms of apparent increase of glutamate after functional stimulation. They need to be taken into account. It's a poster number is 2.
Thank you very much for this.
Hello. Oh.
I think Steve had a question.
Sorry.
Yes, a very interesting talk. Do you have unsuppressed water data?
Yeah, we collect unsuppressed water data, and
okay, so you could see if you could detect a Bold effect.
Yeah, at the beginning of our project, we checked the Bold effect using this unsuppressed water data.
Okay, so you can detect a Bold effect.
Yeah, it wasn't very obvious that we can see it, but after harder analysis, so we detected.
And actually, if I might just give a comment to Ivan, because we have a model for fMRS, and what we say is that the glutamate signal goes up because you empty the vesicle. So we think glutamate in vesicles has very low NMR visibility. So it's not that it goes into the cleft, because Ivan is right. The concentration in the cleft is minute, but you actually release it from the vesicle. So glutamate goes invisible to visible. But that's just a hypothesis. We haven't proved it.
Thank you very much. This could be helpful for us.
Hi, I'd like to make a comment in the context of your presentation, but really in the spirit for all of us at this conference. That I think if you noticed, you got a lot of input and feedback just now. I believe it's because the question that you're asking is very interesting. And the fear that comes up in me and maybe others in this room is that this will not be rigorously checked out before it gets published. And it will be so quickly grabbed by the field, the larger community, that I urge you, beg you to please just have conversations with as many people as you can in this workshop to see if you can vet out each of these questions that they're asking you, because when you publish this, people will believe you.
Okay, so time is going on, so I should start our afternoon session. So my name is Ivan Kaj, and my colleague is.
We now have from Baltimore.
And so my pleasure is to introduce Michal Považan, the first speaker, and the topics of his talk is quality control and pre-processing. Michal.
Thank you very much for the introduction. I hope you had a nice lunch break and are ready to listen to a little bit more of our talks about MRS. So firstly, we should probably answer a question: what is an artifact? It is something that is seen in our scientific experiment or study that does not exist naturally, but has been caused by the way the experiment or study is done. So I will start off the first slide or the second slide already with a take-home message, and that is, it is much easier to have data without artifacts than to remove the artifacts. It is not a new topic. This is seminal work from Roland here about proton MRS in brain and about all the different artifacts that you can acquire during such an experiment. And one thing that definitely drew my attention when I first read the paper many years ago was this educational example about the conspicuity of MRS artifacts as compared to MRI artifacts. So on the left-hand side, you have an MRI of a person that was crawling out of the scanner. Quite dramatic moment, right? And you see that the MRI is corrupted. Even a layman would say so. However, on the right-hand side, you have two spectra. One spectra was acquired while the person was crawling out, and the second one was acquired without any motion. And there is a slight difference in SNR, but even an expert would probably accept the crawling out spectra as an okay spectrum that can be included in the study. I'm an experimental physicist, so I said I would like to see if this is just an educational example or if it can be replicated. So I did a very similar experiment quite recently, and as you can see on the left-hand side, again, it's an MRI of a person crawling out of the scanner. It is wrong, something is wrong with it. And on the right-hand side, you have a crawling out spectrum on the top and a no motion spectrum on the bottom. And yes, there is something suspicious with water and lipids, but if you focus on the part that we are mostly interested in, the 4 to 2 ppm, it is a perfect spectrum, and we would probably accept this spectrum as being completely fine. So it's not just an educational example, it's a real world problem that happens every time when we are carrying out a study. I just have to say that this talk will cover mostly brain proton MRS, but the problem of artifacts is common in any MRS, being it X nuclei, being it body MRS or proton MRS of the brain. There are certain steps how to avoid or how to handle the artifacts during the acquisition or even before the acquisition. First step when you're starting your study is to choose a proper tool, a good combination of your MR hardware, of a scanner, and a software, being it sequence. You should definitely pick a trusty, reliable MRS sequence. You should talk to the community, and thanks to the recent efforts with standardization, there are definitely more options these days than there were, let's say, 10 or 5 years ago. You should also take good care of reading all the consensus papers, all the guidelines, especially if you're not really familiar with the MRS. Second step is getting acquainted with the MR spectrum, getting to know your sequence and what kind of spectra it can produce. Basically learning what is a good spectrum. That takes practice, of course, but it is really crucial when you are carrying out an MRS study. During the acquisition, there are certain steps that you should pay attention to. I will cover them in the following slides. And the fourth step is basically happening during processing of the MRS data. And hopefully that will be covered by Nuno here, who will talk a little bit more about employing the quality control and removing the artifacts exposed. There are certain nuisance signals in proton MRS and in other MRS methods as well that we want to, most of the times, avoid. We don't want to have them inside our data. For proton MRS, it's the signal of water and lipids, water resonating around 4.68 and lipids around 1.3 ppm. And we see that in all the tissues, we get a very similar pattern of having a very strong water signal and very strong lipid signal. In brain, in healthy brain, we should not have any lipids. However, this is very often the case. If you're scanning close to the skull and there is some problem, we get a lot of lipids leaking into our data. I wanted to mention carbon MRS because we're running many carbon MRS studies. And there, the water is not really a problem. We don't really see the water there, but the lipids are populating most of our spectra. And we are interested in most studies into this very minute peak of glycogen. So you can imagine if you have especially studies with higher BMI patients, this can be a big problem, distorting the baseline and really complicating our quantification. That's why Professor Klomp has always been an evangelist of phosphorus MRS, because there are many problems with phosphorus MRS, but at least in terms of contaminants, nuisance signals, we don't have the signals of water or lipids. I wanted to mention something that Georg already very nicely drawn in the morning, and that is the localization in single voxel and in MRSI is imperfect. And when you're planning the voxel on the scanner, you see a nice rectangular box showing your localization, but that's not how it actually is. It's more of a smeared box, and on top of that, you have chemical shift displacement errors. That means that the metabolites are coming from slightly different regions. So this all needs to be taken into account when you're actually planning or positioning your voxel. MRSI, as Georg mentioned, has this problem of a point spread function, and is actually a very serious problem, causing in brain to leak the lipids all the way into the center of the field of view, even though the lipids are far away, somewhere in the skull. And this needs to be taken into account already when you're planning your MRSI sequence. Let's have a closer look on what can go wrong during the actual scan. So you start off with voxel planning, and you should definitely consider where you are placing your voxel, how close it is to the tissues that can actually contaminate your signal, being it liver close to the fat tissue, or the same thing with the motor cortex being positioned very close to the skull. So you need to definitely bear this in mind when you're positioning the voxel. Second, and that was already mentioned as well, is something which we call voxel orientation, or sometimes it's called a slice order. And that is, I think it's something that is very often omitted when you're planning your studies. That is the actual orientation of the voxel and the actual the actual playing out of the sequence. And here, as you can see, there's a big difference between the sagittal, transverse, and coronal orientation, with transverse being or having a really huge lipid artifact, whereas sagittal and coronal are relatively good. And then there is a huge topic of B-node shimming and R power optimization, again mentioned in Georg's talk. And we don't really have time to cover this, but most of the times either vendors or sequence providers give you a solution that you should just follow. But this is not really typical for any other modality, right? If you're familiar with, let's say, just anatomical scanning, MRI acquisition, then you wouldn't take care of the B not shimming or half power optimization. Here, it's absolutely crucial if you want to have data without any artifacts. If we come to an MRS scan, I would like to stress out the importance of having a monitoring of the spectra during the acquisition. So some sort of a live view. Again, most of the vendors provide a solution for you. You just need to open the box and see the spectrum in real time, because that can save your time and the time of everyone who's involved in the study, because I could already see on the live view there is something fishy going on with the person crawling out of the scanner, whereas the final result was kind of conspicuous, right? The question of a good spectrum versus a bad spectrum. So the MR spectroscopy is known to be quantitative, but the quality control is often qualitative. And you can argue, well, we have line width, we have SNR, we can judge the quality based on the nuisance signals. We can create some score and rule out the spectra that have higher score than a certain value. That is all true, but at the same time, at the end of the day, when you have your data, you will need to decide, are these spectra good or bad? It will not be a number. It will be included into the study or not. And for some, it is quite easy, like this one on the left-hand side. You can say it's probably a good spectrum. For the others, it will be easy to say it's a bad spectrum. Whereas for some, it will be quite difficult to say if you don't know anything else about the study or how the actual spectrum was measured. So you need to know all about your data, how it was acquired, what was the shim, if there was any problem with RF shimming or the power, and so on and so forth. Coming to a second part of my talk, I'll just mention some of the common artifacts that you can encounter when scanning with proton MRS. First being so-called spurious echoes and ghost signals. They have a very, very typical, very quintessential pattern around the water resonance, but very often leaking all the way into your spectrum. And if you see the individual traces, you can see it's quite a dramatic artifact that you can get. It's affecting the static field gradient volume selection methods, that is steam, press, S-laser, spatial, being it single voxel or MRSI, doesn't really matter. And it's mostly caused by the incomplete spoiling due to suboptimal sequence or just due to some other problems such as motion or B-not inhomogeneities. And there are many ways how to handle this. So you can start with actually optimizing your sequence properly, as Carl mentioned some time ago, using the dot cops. You can employ gradient reversal. You should definitely employ phase cycling, but that's quite a common thing to do. For us, in many studies, it's helpful just to enable the OVS that will crush the signal from outside of the voxel, because the spurious signal is actually coming from outside of the voxel. It's never from inside of the MRS voxel. It's coming from outside, and it's mostly the water signal that is causing this. And there are also some post-processing methods that handle the spurious echoes, but they are not really trivial to use or implement. On the second place comes the B-not field inhomogeneities. And this is a problem, especially for MRSI, for higher spatial coverage, because we know due to natural line widths and due to the constitution of our body, we have just various line widths across our tissue of interest. And if it's not handled properly, the MRSI is rendered completely unusable, right? It's just very, very difficult to get anything from badly shimmed data. But same applies in case of some susceptibility effects or implants and single voxel. These data are provided by Peter, because he said he's an expert on bad data and acquiring bad data, because he's been in the field for such a long time. These are, and as you can see, these are basically data that can't be used at all, just due to the dental braces or surgical staples and implants that were placed in the subject. Insufficient B1+, it's often a problem at ultra high fields. This sequence, or this is an example of a study that we've carried out in motor cortex, and we knew it's an S-laser sequence. And we used the sequence previously in ACC. And we applied a sequence with identical parameters in motor cortex. And we saw there is probably some problem. We're not really sure what kind of problem is that the SNR is lower, and then the ratios of creatine, choline, and NAA are different. And thanks to the fact that we are just paying attention to the pre-scans, we already saw during the acquisition that the RF power just could not be reached. And that means that our data had to be thrown away, and we had to think of a way how to actually acquire data with sufficient power. You can try and change the sequence parameters. Yes, that works sometimes. You can try and use dielectric pads. That's actually something that worked for us. So you're just placing the dielectric pad to change the local B1 plus, or you can employ some RF shimming methods, but that's again quite advanced. Coming to a fourth artifact, it's lipid artifacts, right? So we already mentioned that in proton MRS of brain, we should not have any lipids if it's a healthy tissue. However, oftentimes we have such huge peaks of lipids that are completely destroying our spectra and quantification. Excuse me. And the cause to it can be a voxel misplacement, can be very often is subject motion. Or in MRSI, it's just a matter of the method, right? So in MRSI, it's very common to see lipids every time when we scan, and it doesn't necessarily have to be a problem. We can employ various ways how to tackle this. Either we do some sort of a lipid suppression or handling, or we again try to handle the lipids in post-processing. There's a different approach to lipids in MRS and in MRSI, because in MRS, your sequence performance is determined by the performance of the RF pulses. So if the RF pulses are performing well, if your sequence is working properly, you should be fine, excuse me. You should be fine with just using the actual sequence and maybe adding the OVS. Whereas for MRSI, it's all complicated with the PSF. There are many methods how to deal with this. Apologies. Being one of the things that Georg already mentioned is an OVS. So basically placing many slabs on the skull just to suppress the lipids. However, this is quite SAR demanding. And then there are inversion recovery methods where you're losing quite a lot of signals. So this is also not the perfect solution, but many times and in many studies, this works. Another option is to use just dedicated hardware, such as crusher coil. But for us, in many studies, it worked a combination of some kind of a reconstruction, filtering, regularization with L2, or PSF improvement just by increasing the resolution of your MRSI. Coming to the last group of artifacts that I'd like to cover, it's motion-induced artifacts, and they come in different forms and different shapes, but often they are connected to the alteration of the sampled region, which means basically that you are acquiring your data from a different region that you planned. It comes with the change of an F naught of the transmitter frequency, change of the main magnetic field of the B naught, and change of the RF field or the B1. So basically, all the artifacts that I mentioned so far can happen due to emotion, and this is a huge problem for any spectroscopy. What is a remedy? Well, you can tell the subject not to move, of course, or you can try to immobilize the subject somehow, or you can, in post-processing, remove the motion corrupted data. This works quite well. I'm sorry. But we can also employ motion correction methods that I'll cover now in the following slides. In general, the motion correction can be divided into retrospective motion correction that is done in recon, and a prospective motion correction that is done during the acquisition, during the actual acquiring of the data. The correction needs to focus on these three things. You should do a positioning update based to correct for the localization errors. You should do the frequency update to correct the frequency errors, and you should do a shim update to decrease the field inhomogeneities. Sorry, to correct for the field inhomogeneities. On top of that, you can decide that you will reacquire the data if the motion was too big. So let's say if the motion was bigger than 1 millimeter in one direction, you decide to reacquire the data again. And of course, that takes slightly longer, but sometimes it's a lifesaver if you have subjects that are moving more in the scanner. The positioning update is usually carried out using either internal or external methods. Internal methods are navigators. So those are very fast MRI sequences that can give you a position of the head. Every TR are usually placed before the actual MRI sequence. Or you can use external navigators such as optical cameras and NMR pros, and there are multiple methods using either some sort of a marker that is placed on the subject or that are markerless. I'm very I'm very sorry. There's a Lausanne air getting on me. That is the positioning update. And then we can do a frequency and shim update. We can either do a shim update based on the navigators. So some navigators can actually create very simple B-node maps. And based on these B-node maps, you can update your shims. Or you can use 1D projection methods such as FastMap that can be interleaved with your actual MRS sequence. But then there is a problem with these projected methods that they are suitable only for smaller volumes, that is only for MRS. You can also do something like a global FID or a water scan that is interleaved with your sequence. That was mentioned by Kim in the morning. That is very popular in MRS acquisitions because you get a very fast and very precise update of your center frequency. So coming to an end, finally, I will just try to draw your attention to some of the sources, definitely Roland's paper, then all the consensus papers as well, and a very good review paper on motion correction by Muhammad. And just to summarize, again, I will repeat it because it's very important. It is much easier to have data without the artifacts than to remove the artifacts. Second, learn how does a good spectrum look like for your particular sequence. And third, focus on the pre-scans and live monitoring, because if you avoid having any artifacts in the spectrum, your life and the life of all the spectroscopists that will quantify your data will be just much easier. Thank you very much.
Thank you, Michael, for your very nice presentation. And I have to apologize for my wrong introduction of your talk. I read the title from the next talk. So the original title was Artifacts and Correction for MRSI. Sorry.
No worries.
Well, so we have time for some questions.
Hi, Michal. Thank you very much for the nice talk. I have a question about the physiological artifacts. So artifacts induced by the scanner. For example, you can have PNS activation due to the scanner, and this PNS activation might modify some levels of neurotransmitters or metabolites. How is this? How do you deal with that? Is there a method, or should you just discard, or is it even important?
Well, I think in general, you should try to avoid having a peripheral nerve stimulation during your acquisition. It's especially the case when, especially the case when you are doing some studies that can, that are looking at metabolites that have something to do with neurotransmitters. But I think most of the times the participants experiencing the PNS, they just complain, so they let you know. So you're avoiding this by just asking the participant if everything is working out fine.
Is there a procedure that is usually used, like asking the participant to say something openly, or you are hoping that the subjects will raise the hand?
Well, the vendors provide, there is a pop-up box. Every time when the PNS reaches certain level, there's a pop-up box that will tell you that there may be a PNS. There may be a PNS happening. So you should definitely talk to the participant before you start the sequence and inform him or her that this can happen. Because it is very much depending on the actual participant and on the body size, and so the fact that the pop-up box shows doesn't mean that it will that the participant will feel the PNS.
Thank you.
Okay. One more question.
I just have a quick question, pretty much for everyone, not just for you. You mentioned, first of all, great presentation. Thank you so much. The question or request is, you mentioned patient immobilization. We learned that like putting sponges around the neck to tilt the head helps with lipids. Is there anything else anyone else recommends to get a better data? That seems like an easy, low-hanging fruit. So if anyone does, please let us know. For trainees, we would love to learn that.
So from the last MRS workshop in Utrecht, I remember Christoph Buchem is here, mentioned that you should do verbal slapping first. So tell the participant not to move when you see that he or she moves, and then comes the real slapping. But of course he meant it he meant it as a joke, but I remember it every time when we were putting the subject in. Well, there are, to be honest, I'm not a big fan of padding the participants too much, because I know myself that when I go inside the scanner and I'm padded all around, that I start to move even more, because I just don't feel comfortable. I think it's a combination actually of debriefing the participants and really stressing out how important it is not to move, and also placing certain padding around the head in case of a head scan, of course. I don't really like any extreme fixation, to be honest, even though it's sometimes quite common. You can sometimes try to tape the nose to the front of the head coil, or different methods, but yeah, I'm not a big fan.
Well, thank you.
Just a
only
quick
quick
comment.
question
Regarding this, my experience is that if you want to limit movement of your patient, especially for long excision, something that has been done for functional imaging in general, is to have a crossbar or a fixed image to give to the patient. This will fix his line sight and will limit his movement. At least from my experience, it helped with and without to limit the rotation, especially of the head. Okay.
Okay. Thank you, the speaker, once again. Thank you. And so it's my pleasure to introduce the second speaker, and it is Nuno Barros, from Ecometrics, Belgium. Yeah, and the topic is now quality control and preprocessing.
Can you hear me well? Yeah. So a little bit challenging to cover preprocessing and QC in 20 minutes, but I'll do my best. Thank you for the introduction, and also thank you for inviting me to be here. It's really a pleasure to see many of you, a lot of familiar faces. Yeah, so let's just get started. So overall, we have to start by looking at the context of preprocessing and QC in the MRS processing workflow. And of course, that what will be optimal for each case, it will depend both on what comes before and what will come after. So what type of data you are acquiring and all the characteristics of this, what is the quality of this data, and what will come after, what quantification method you're using, if you're doing some post-processing methods, if you're doing some, what is the results interpretation going to be like. And I try to look at this in very general terms as a way to decrease the information noise. So here I'm not talking just about the noise that you get in FID, but all the type of information that you collect that in fact will not be used for the end result that you want the spectrum. So you do all in the acquisition, or they try to minimize the signal noise as much as possible and collect the information that you really need. And in preprocessing, you try to lower this down, but of course, you need to start with reasonable quality data. And as the pipeline, as you proceed in your pipeline, you start to put this data into context and extract the information that you want to answer your research question or yeah, to help understand better what is going on with your patients. So I will cover some of the most common preprocessing steps. I'm sure many of you already worked with many other steps that are not here covered, but I would say that these are some of the most common steps, and try to give a very simple overview of them, understand, explain why they are used, what is the motivation to use them, and some characteristics that we should know about them to help you define your pipeline. So signal combination, the main motivation here comes from the low metabolite SNR that makes it that we acquire different acquisitions. So we repeat the same sequence several times, and also collect information from multiple coils as a way to increase this SNR. Of course, these signals, they are quite separately, but very often, if you don't change the settings in the scanner, they will already be combined by the time that you have your DICOM file, if that is the way you work. So you have to make sure that you store the data separately. And this comes with different challenges, namely the spatial sensitivity of the coils. So each coil will see a specific region. You also have the correlation between the different coils. So ideally, you would like the noise not to be correlated so that it all cancels out, but that's not the case. The noise is correlated between the different coils. You also have phase differences between these coils that should be modeled. And the phase of the water and the metabolite peaks might also be different, which can lead to some cancellation artifacts. There are many methods to do this, but I would recommend this article that this overview article that Miriam prepared from the group of Sarah Nelson, who left us very recently, someone that I'm sure many of you knew well. And what you can see here in this figure from this paper is that first, one of the main impacts is the number of channels. So you have here SNR from NAA from 32 channels and 8 channels, and then different ways of doing this coil combination. And you can see that the biggest jump comes from simply using more channels. Another interesting thing is that the choice of the method has a bigger impact when you use more channels. Then you'll have a higher variability there. And some of the messages from this review paper is that when residual water is present, it's a good reference for determining phasing and channel weighting factors, and that phase corrections should be done voxel-wise. Moving to the next step, also very common, but not always done because it requires a separate scan, a separate water unsuppressed scan. And here the motivation is that eddy currents produced from the rapid gradient switching will create time-dependent frequency offsets, which will cause line shape distortions. And here you have a nice example where you can see how this can affect the line shape. And the way to do this is exactly to understand in that unsuppressed water scan, how is the phase evolving in time. And once you understand this, then you can basically correct all your peaks in the water suppressed. I would recommend this whenever possible. This is sometimes included already in the quantification software like NLC model, Fit8 and Tarquin, but it's not always possible. It requires separate acquisition. Residual water removal, also one of the most common steps done, and one of the steps that has the biggest impact, because as we all know, the water concentration is way higher than the one of the metabolites we are trying to measure. And the water suppression in most of the times is insufficient. So you still have a residual water peak that in many situations you just cannot ignore. And this will impact different preprocessing steps and your quantification. So it's better that you get rid of it. And the most common approach is simply to find a way to fit this peak and subtract it. And the most common fitting method, there are several, but HLSVD is one of them. And here I did one experiment for you to understand the different scenarios that you have and why you need to do residual water removal. So if you look just at the water unsuppressed spectrum, and we look at the region of interest where most of the metabolites are located. So here I'm just staying on one side of the water, I'm not going to the other side of the water. We can see that the quality is quite terrible when you apply HLSVD, and the residuals that you have still from the fitting, they are quite dramatic. They are at the same order of magnitude as the other metabolites. So you cannot do much with this. Whereas if you do good water suppression, you are left with a relatively small water peak that can be easily removed with HLSVD. So then you are left only with the metabolite peaks. So here I'm showing you two scenarios. The first one, where residual water peak is removed, is applied to unsuppressed water spectra. And this generally provides very poor quality spectra. And this has to do with the limited resolution of your analog to digital converter, and that small fitting errors of such a large peak will be already huge. So it's definitely, no one would do this. Effective water suppression, here if the method works well, the residual peak might be relatively small and not impact so much what you're trying to do next with the spectra. Still, I would recommend it, but I would, in these particular cases, I would not say that is absolutely necessary. It might depend on your quantification method and and the tools that you're using after. But in many cases, your water suppression doesn't work that well as we saw in the previous case, and you end up with a huge baseline coming from this water peak, and this will have a dramatic effect in your quantification approach. So you definitely need to do some water peak removal in these cases. So in general, I would say that it's always perform residual water removal. I think it's the safest approach. And another thing that it's interesting to see is that it will not only impact quantification, but also, for instance, in this example where we trained an automatic quality control classifier using water removed and water non-removed data, we see that with the data where we didn't use HLSVD, the median is in fact slightly lower. So the median performance is slightly better, but as you can see, you also get a lot of cases really wrong. So I think it's another proof that independent on the application, it's better that you perform residual water removal. So now regarding autophasing, so another basic step that we all are used to do, and here one of the main motivations is that autophasing that was already performed by the scanner software is often affected by the residual water peak. And so when you do this residual water removal, your metabolites are out of phase. And there are two main approaches that are used. One of them is to use the first point of the FID. This corresponds basically to the average of the frequency domain spectrum. Or you can simply maximize absorption signal in a given range. This is a little bit more robust and focus on the signal or on the region of interest. It's important to know that if you have frequency dependent phase deviations, you should apply zero order, but also first order phase correction. In data that I worked with, first order phase correction was not always extremely necessary, but maybe in other cases might be. So yeah, phasing is a simple and essential step. Frequency shift correction, another essential step, and this comes from the B-naught inhomogeneities that will cause frequency drifts. And here you have to understand that it can impact different things. So if we are talking about, let's say, machine learning and we're extracting features from regions of the spectrum, these regions will represent different things if the spectra are located if they are shifted. Or if you are doing quantification and your metabolite models or your quantification method cannot cope with these deviations, you will get very bad results. So it's an important correction that needs to be done. So I would say that when defining the method and the target frequencies that or the ppm values that you want to move your spectrum to, you should consider your metabolite model so that you really bring your spectrum to the, let's say, the space or you align it with your metabolite model. In MRSI, it's important that you not only collect the global shift, but also that you do this in a voxel specific way. And you can do this by trying to bring the mean spectrum to the metabolite model to find the global shift. And for the voxel specific, you can maximize the correlations between the individual spectra and the mean spectrum. So I would call this as an essential step. Apodization. Here, the motivation is that the last part of the FID contains mostly noise. However, the problem is that if your quantification method uses this last part to assess the noise level, this will affect your quantification error estimations. It also will affect the line shape. So very often this is not recommended by some quantification methods. And I would say that it might be recommended for visual interpretation of spectra mostly. Finally, zero filling. Here, I think the main motivation is just that the signal resolution is not as high as you would like, but in fact, no new information is being added with this step. And so you, if you would even go to store the data, you might increase your data storage requirements by quite a bit. And so, I would say that this should only be used for visualization purposes. So now you know your steps. How should you go about setting up your pipeline, or what should you consider? So it's very important that you understand what are characteristics of the data that you have. So the sequence, the scanner preprocessing, what was already done before the files were saved in the scanner. What are the time constraints and the system that you have? Sometimes you want data to be processed quickly so that a decision can be made. Others you might afford to have quite some time to process the data. It's very important that you know the quantification method that you're using. Is it robust to frequency shifts and dephasing? What are the characteristics of the metabolite model? What line shapes does it consider? And what is the end user of your spectra? Is this a clinician, a spectroscopist? What output you want? Spectra, metabolite maps? And then you should understand the interactions between steps. So, for instance, the residual water peak removal should be performed before the auto-phasing, and there are many other interactions like this, and define the order accordingly and optimize for your needs. I think that it can be interesting that you define some key performance indicators so that you can keep track of how good is your processing pipeline, and then you can optimize this, and not only the mean values of your KPIs, but also maybe some percentiles so that you can have a measure of the robustness of your pipeline. And a recommendation for a very, very nice overview over the steps comes from another consensus paper that I recommend for you to read. And as we also saw here today, this MRSHub, this nice platform that has been prepared, also has a section on software and code. So there you also find different tools that might help you to process your data. So now moving to quality control, I would like to start with a practical example. So I'm showing you here two metabolite maps from a tumor patient. one acquired at baseline, another one acquired 4 months after radiotherapy. And here we can distinguish mainly two regions. So the tumor region that looks like it's responding to therapy. So you have a lower choline to creatine ratio, but also a region that has a slightly higher choline to creatine ratio. So maybe representing some tumor infiltration, or maybe not, because if you look at the quality of the data, you could not make any of these conclusions. The data of the second acquisition is really terrible and highly affected by inhomogeneities, and so all that I just said is simply not valid. You cannot interpret this spectra, and so artifacts and poor quality spectra may lead to wrong clinical interpretations, which can be quite dangerous. And very often this data contains artifacts. So it becomes really important that you do quality filtering, especially if your data is being used in clinical routine. You could identify these cases manually, but this is subjective and it will be very time consuming. So if you have sequences like EPSI and you want to go through all the spectra, how much time is this going to take you? And very often it requires an expert. So a clinician will maybe not be comfortable to do this quality assessment. One of the most common ways to do quality control is based on cameral lower bounds. I call it more, I would distinguish here quantification quality control from signal quality control. And so cameral lower bounds fit better in quantification quality control, and are these are used to filter metabolite maps. However, these will only be valid if the model applies to the data. So if you have large artifacts, your cameral lower bounds will not be meaningful. And it's also another nice thing about quality control is that it can help you to remove nuisance signals. And if you have less data, just the core of it just the good signals to work with, this might also help you to speed up processing. So that's also a very nice motivation. So what I propose as a workflow for a quality control would be to start. So here, I'm talking mostly about MRSI data. If you start with your data, you should do residual water peak removal. This just has to do with what I mentioned in the past that will affect the performance of your classifiers. Then you can do your automatic signal quality assessment, and you immediately reject those spectra that are highly affected by artifacts. You proceed with the good ones that are worth the rest of the analysis. You perform quantification, and now you can do some quantification quality assessments. And based on the criteria that you defined, you can then just keep the reliable results. And there you go, you have your metabolite maps. And you can also have some error maps that can support your decision. Here I'm giving you just a practical example of how well this can work in practice. And you can see that it can be quite fast to classify data, so you can get this really fast. What would take a manual operator quite some time to do, and the performance is often quite good. While implementing this, we also did some studies on the importance of different features. So classifiers like random forest help you to understand what is driving the decision. What are the features from your spectra that help you to separate good from bad quality spectra? And typical features that would be used in clinical practice would be the SNR of the main metabolites, but there were also some interesting features that were normally not looked at, like the frequency domain kurtosis or skewness, that especially for long TE are quite relevant. And here you can see why this works so well. So it's just based on the fact that in good quality spectra, for most cases, if your baseline is of course good, then most of the cases will have very low values, and only in the peaks you have this high value. So if you plot an histogram, most of the histogram will be skewed towards zero, whereas if you have just a very noisy spectra, you have values all over the place. So yeah, in time domain, you also have some regions of the time domain signals that are more relevant. And this just has to do with the faster decay. So with the faster decay of the bad signals, of the bad quality signals. More work was done by Fatima Tensawiti that used not only the features that were used in the past, but also features extracted from single MR from Siemens. And here what was interesting is that, so it confirms that frequency domain skewness and kurtosis are indeed important features, but in this particular case, it was identified that the residue that you have in the lipids, so between the fit that Siemens performs and the lipid signal that you have, is one of the most interesting features. One of the main drawbacks of all these automatic classifiers is the time that you need to label data so that you can get a high performance classifier. And so work was also done in the past to try to understand how to speed up the labeling. And here I'm plotting some spectra, and you can see that they are separated between good quality, accept, and bad quality, reject, and ordered by two of the most important features that help to visualize and group the spectra by quality. And the color of the spectra represents the probability given by the random forest of giving them, of accepting the spectra or not. And what you see is that what you're trying to do in, of course, the boundary that what you're trying to do is to separate these two groups. So you're trying to find a multidimensional boundary between the two spectra. And if you just look at these two features, the information to define this boundary comes mostly from this. It doesn't come from those very, very good spectra that you might be labeling over and over just as a repetition. What you want is really the spectra that are difficult to label. And so we defined a way to identify the spectra. So you can have a first random forest that classifies this. And from that probability, you just select the ones around 0.5 if it's a binary classifier. And then you can iterate. So you can keep on selecting the uncertain cases, labeling those, and training the classifier, so retraining it and labeling more uncertain cases and proceeding like that. And this can help you to save a lot of time. So if you do standard labeling, I would say that in many cases, I would recommend up to 10,000 spectra. Of course, it depends on the application. Active learning, we can get maybe similar performance, just labeling a fraction of it. Other advances in this field that I find quite interesting is that using deep learning, you can then look at the regions of the spectra that mostly influenced your decision, or mostly influenced the decision of the classifier to define if it was a good or a bad spectrum. And this is, I think that's explainable AI is a trend and an important one, because we want to understand why we get the results that we get. And so I think this is very interesting. And another interesting result coming from this group, from Gurbani, is the fact that if we look at the CNN and the average of the human raters, we see that in fact we can understand AI as a tool to create consensus, not only to really do the task that we are asking it to do, which is to label the spectra, but maybe also to bring people together and different ideas of quality. And I think it's what is very difficult sometimes to convey in words, to find long protocols of what is a good or a bad spectrum. Maybe these tools can help us to get there. So to finalize, I would like to give some of what I think could be some future directions in quality control. So in terms of labeling, I think there's still a lot of work that could be done here to reduce the labeling requirements. There's a lot of repeated information, and there are tools like NMF or PCA that could be used to see what identifies spectra that are very different, and so that you're not always repeating yourself to the classifier, let's say. Definition of consensus guidelines for labeling data quality could also be interesting to explore. I think each center has a different idea of what they think is a good or a bad quality spectrum, and labels from multiple experts from different centers could then be used to build this consensus classifier, which I think could be an interesting project to do. Partial rejection. So if you looked at the previous plot, sometimes we have spectra that have regions that are bad quality, but other regions, in fact, we could still accept and do something with it. So I think that could also be something that would help us to not reject so much data and still keep some reliable portions of the spectrum. But that would require that we are able to predict quantification error in the presence of artifacts, which will come with its own challenges. Assessment of QC performance for different types of artifacts could also be interesting as a way to help us understand what are the main bottlenecks, what are the main challenges for these classifiers, and how to fix them. And I would like to finalize that whenever possible, not only we should detect, but if possible, fix them. And coming from the work of Prithvi and Roland Kreis, I think it could be a very interesting direction to pursue, to have ways of fixing many of these artifacts. And so with this, I would like to finish and acknowledge the previous teams I worked with in Bern, and also the Transact it's already a few years ago, but still still present for me, and my team at Ecometric. Thank you all.
Thank you, Nuno, for your nice presentation. Well, we must move on because of the time requirements. So thank you. And I will just repeat his words, what he ended. Well, if we have good performing sequences, it's always easier for processing, not to be forced to remove some artifacts, but there will be artifact-free data. Thank you. And now I will move microphone to my co-moderator to follow.
Yeah. The next presentation is about fast prospective motion correction for spectroscopic acquisitions from Isaac Adanyego, sorry. Yes, from University of Minnesota.
Thank you. So unfortunately, Isaac was not able to make it, so I will present the talk. So subject motion during MRI and MR scan is a major issue, especially in movement disorder, children and elderly subjects. So as shown in the previous talks, subject motion during MRS will affect the SNR. You can have lipid contamination, you can have noisy spectrum and a broad line width. And also you might be acquiring data from the wrong voxel of location. So there exist several prospective motion correction techniques. We have external devices, which are optical camera field probes, which are expensive and need additional hardware and software. And you need to integrate these systems with the MR system. And you also have image-based navigators, such as the volumetric API, the Promo, the FID, and MS Space. These are less expensive and doesn't need any additional hardware, but they need to be implemented into your sequence. So previously, some of the sequences, the time taken between the navigator and the MRS sequence was quite long, which was around 1 second or more. So the aim of this talk was to develop a fast image-based motion navigator, which will minimize the time between the start of a motion and the start of an MRS acquisition. So the protocol was based on a multi-slice to volume registration, which was shown by Honkis and Porter in 2018. It was based on API technique, and we implemented the same on Spiral. So basically, at the start of your sequence, you will select your brain region, usually the whole brain, where you will adjust the first and second order shim, which I'm referring to as the whole brain visual shim. Then you will acquire, let's say, several 30 slices, actual slices, to cover the whole brain. And this will be our reference volume. So for my study, I used 30 slices, and that's the parameter I used. And in your MRI sequence, you will acquire select 3 slices within the reference slice, which are also axial and will match the parameter of what you use, like T and TR. And you will acquire 3 slices with a whole brain shim. Then you will use these 2 data set to perform multi-slice to volume registration in order to get the free translation and free rotation. And this is done in the ITK library on the scanner. When using the motion parameter, you can update the RF pulse and gradient. Then you will perform the B0 dynamic B0 shim correction, just the linears and the frequency, and you will execute your S laser sequence. And this is repeated for the next year. So the motion navigator was based on spiral navigator. We use the spiral in and out trajectory to minimize off-resonance effect. And the real-time shim correction was based on a 2D RF low flip angle pulse, which we will adjust just the linear shim inside the voxel. So we did not adjust the second order shim. And we also have a voxel localized steam sequence, a low flip angle, to measure the frequency and correct for the frequency so that we have a water on resonance. So we scanned 5 healthy subjects on a 3 Tesla scanner, and we modified our semi-laser sequence, where we replaced the vapor module with a metabolite cycling pulse in order to reduce the time between the motion navigator and the S laser acquisition. But we still kept the free OVS module. So our voxel of interest was in the prefrontal cortex, which is very susceptible to motion due to the close proximity to the sinus. So basically, at the start of a study, you will define your reference volume shown in the yellow box. Then in your MRS sequence, you will define the, let's say, the prefrontal voxel here shown. And the sequence will automatically select 3 slices such that it doesn't go through your VOI. And we also compared 3 conditions. We considered the baseline where the subject did not move. The subject moved, but the MOCO, shim, and frequency was disabled, which is denoted as NOCO. And the shim MOCO where the subject moved and we perform the motion, shim, and frequency correction. So usually when you do single voxel, you will perform B0 shim in your voxel instead of a whole brain as we are doing right now. So we wanted to compare the line width, what line width we are getting using just the whole brain shim. And the second one is whole brain shim, and we are adjusting the linear shim inside the voxel, or the voxel-based shim where we are adjusting the first and second order shim inside the voxel. So we measure the line width in PFC, PONS, and PCC. So here's the result we got from 1 subject. So that's the spiral navigator we obtained at the first TR, and that's the baseline spectrum. And when the subject move, and in this case, we ask the subject to rotate their head to the left, I would say, which is, I think, a Z rotation. You see that it's much broader spectrum, and also it's not clear here, but the intensity of total creatine and choline is changing, showing that you're measuring from a different location. And when you have both shim and motion correction on, you can see we are getting back the same spectral pattern and line width as with baseline. And that's the SP nav images acquired at the end of the scan, which you can see after correction. So here's an example from 2 other subjects showing that that's the baseline spectrum. That's when the subject was asked to rotate their head. You can see it's degraded, and you got back the same spectrum compared to the baseline when you have both shim and motion correction on. So we also compared the line width. So if we start with a PCC, if we use the branch shim, which is you're adjusting the first and second order shim through the whole brand, compared to whole branch shim plus linear or force map, you get comparable line width because the voxel is central to your is centrally located. But for PFC and PONS, you do see if you're just using the branch shim, you're getting line width greater than 10 hertz. And just correcting for the linear, on top of a branch shim, you're improving the line width a lot. For PFC, you're getting less than 10 hertz. And PONS, it's around 13, 14 hertz. And it is, the values are within the recommended range from the MRS consensus paper. And if you use voxel base, which is force map, you see that it's much more, you're getting better line width compared to the branch shim plus linear free. So for this study, we used 3 slicers in the MRS sequence. So we wanted to compare how many slicers, minimum number of slicers you can use in order to have good motion estimation. So we took a reference data from one subject and applied some translation and rotation in almost all directions. And here's how the subject is rotating their head in simulation. So we compared, we registered all 30 slices. We were starting reference image, or we selected 1 slice below the voxel, 2 slices, 1 below and 1 top, 3 slices, or 4 slices. And all the registration was offline. And here's the difference between the imposed change and what we are measuring. So basically, if you use at least 2 slices, you can see that the translation is within 0.02 millimeter, while and rotation is within 0.05 degrees. But if you use 1 slice, as shown in the blue one, it's much higher. So basically, I would say this plot shows is what we expect for the lobar bound using this technique. So in conclusion, we showed that using 3 slicers, we can get fast and reliable prospective motion correction for single voxel. And we can further reduce this time if we use 1 or 2 slicers. So this technique is not limited to single voxel, but can also be used for MRSI acquisition. Thank you.
Thank you. If you have questions, you can take them.
Thank you very much. This is looks very promising. Of course, if we could measure at a steady position. The shim is probably not immediately co-registered. I mean, if your motion is only very small, the shim would be okay. If the motion would be larger, do you think you get a problem?
Yes, you will get a problem depending on how much the subject moves. So usually that's why in each TR, we are applying the B0 shim. So if a subject is not moving, the changes we will see will be minimal.
Yeah, well, but it's very nice. Thank you very much again.
Thank you.
Okay, if there's no further questions, let's thank our speaker again. And the title of the next presentation is Deep Learning Pipeline for Quality Filtering of MRSI Spectra, given by Mladen Rakic from Icometrics, Belgium.
Hello, can you all hear me? Okay, good. I much prefer the view from up there, I think. I also want to thank you for this clip-on microphone because I'm known for not being able to hold a microphone properly. Last time I held it too close, I sounded like a pilot announcing things in an airplane. Anyway, I want to talk to you today about deep learning pipeline for quality filtering of MRSI spectra, and it will relate a lot to what Nuno was saying in his presentation. Before I start, though, I would like to make some acknowledgements. This was the work that I did at University Hospital Bern in the lab of Hans Lotmo. It was my 3-month secondment there, and I would like to give special thanks to the co-authors, Hans and my main supervisor, Diana Sima, and students from University Hospital of Bern, Federico, who is not here today, and Guodong, who is here today somewhere. The motivation for this work was the rise of these novel 3D MRSI acquisition protocols that capture a large number of spectra, counted in thousands for a single patient, as you may be familiar with. And that's a lot of spectra which need to be processed and quantified, but in reality, a lot of these spectra are not even worth processing. So the question is, what can we do about it? And can we filter out the bad quality spectra automatically and quickly? And what I mean by that is, here are some examples of different classes that we defined. We went with 4 different classes in this particular research. On the upper left, you see noise. The upper right is a spectrum that's highly impacted by lipid contamination. The bottom left is a sort of good quality looking spectra with a minor lipid contamination still present there. And the bottom right would be a good quality spectrum. And you will see some descriptions now on screen, and don't take these too literally. But for the purposes of classifying and consequently quantifying afterwards, we can say that noise spectra are useless for quantification. The highly influenced lipid contaminated spectra are somewhat useless. These are potentially useful spectra. And lastly, we have useful or good quality spectra. So the idea is to come up with a supervised deep learning solution for classification of spectra based on the quality into these 4 classes. We have a data set, which is around 23,000 twice manually labeled spectra. And Nuno was talking about how time consuming that is, and an expert in this room knows how time consuming this is. And it's spectra coming from a single subject, so it has a rather unbalanced representation of the 4 classes. You might imagine there's a lot of noisy spectra and a lot less of these potentially useful spectra, for example. The data was acquired with an EPSI variant with a novel type of spectral editing named SLOW at ultra high field of 7 Tesla MRI scanner. You will hear a lot more about the SLOW technique from the authors of the SLOW. Guodong will give a talk about it on Wednesday. The whole brain editing can be obtained within 10 minutes acquisition time. And water lipid suppression is implicit in SLOW editing, so no HLSVD was necessary. I will, however, talk about the deep learning architecture, which was a rather simple ensemble network. It's a 2-part ensemble network. So I used autoencoders for feature extraction, automatic feature extractions from the spectra, and then a simple classifier for labeling the spectra into the 4 defined classes. And then I used 3 pairs of these autoencoder classifier networks, which were trained on different parts of the spectra. So 1 pair on absorption, the other on dispersion, and the third one on both absorption and dispersion. And why, you might ask? Because ensemble classifiers prove to yield better performance because of the more robustness and the added value of majority voting. This is a very abstract overview of the architecture. So as I mentioned, we have part 1, which is convolutional autoencoders, which serve to extract features in this compressed, well, if you will, it's a network that takes an input and tries to reconstruct the same thing at its output. It does so by compressing the data, so it searches for the most useful features in the data given at the input. And so this middle layer is compressed representation of the spectrum. And then I have a second part, which is a classifier, which takes, so the first part of a classifier is untrainable, and it's the same as the autoencoder. So the first part of the classifier serves to extract features and then has an added added layer for classification into 4 classes at its output. And then a final classification label is a majority voting of the 3 pairs of the classifiers that I explained before. I'm going to give you some numbers. We have 4 classes previously defined, 0, 1, 2, and 3 here. And I'm showing you here the F1 scores and how good the classifier performs based on the ground truth or manual labels. So in this confusion matrix, we have true labels are the ground truth, the labels from the MRS expert. And every column are the predicted labels for each of the 4 classes. So in an ideal world, we would expect only values on the main diagonal, meaning that the model is perfect. All values outside of the main diagonal are mistakes made by the model. But I would argue that given the amount of spectra that we have, these mistakes are not so grand. So you can see the performance for 4 classes, and the lowest one is for class 2, which was the potentially useful class, the spectra, which are slightly impacted by lipids and macromolecules. But it's also, that class has the lowest number of spectra in the data set, if you will. Here are some qualitative results. This is how it looks like. It's a map where blue is the noisy spectra as predicted by the model, and the red are the good quality spectra. And you have a small selection on the left, and the average of that selection is a spectrum shown on the right. So it looks okay qualitatively. We expect to see highly lipid impacted spectra around the skull area and good quality spectra inside of the brain. This was evaluated on a healthy subject, and the spectra from this healthy subject were in no way used in training. So this is data that was never seen by the model. Okay. Then we sat down and we said, okay, what can we do to improve this? Because if I already mentioned that we have data coming from one subject, and that's one healthy subject. So how good would the model be when evaluating on an unhealthy subject? And the easiest idea was to add more data into our training set to make the model more robust. And we did that by adding around 12,000 spectra from 5 additional subjects. Those were also been manually labeled with a lot of time. And to somewhat account for the class imbalance and to add more variety into the data set. So the updated model was trained on 25,000 spectra and validated on 11,000 spectra, more or less. And these are the numbers. They're not largely different from what you saw before. I will give you a nice overview table later comparing them. But it's a good thing. They're not much better either. But diversifying the data set did not influence the final outcome is, I think, the takeaway message here. I will show you the updated qualitative results as well. So this is the same slice on the same healthy testing subject, which comparing it to the previous one doesn't look much different. I think there's a slight more nuance between yellow and red labels than before. And if you think that these numbers are maybe not good enough, or that, for example, class 2 only has F1 score of 0.82, for example, why is it higher? I also want to touch upon a thing that was also mentioned by Nuno briefly, which is the intra and inter-rater variability and comparing model results to a human rater and how that bodes in this context. So I told you that the data was twice labeled by the same expert. And I want to show you the similar confusion matrix, showing the labeling consistency of the two separate labelings of the original data set coming from one subject. So again, we have a similar confusion matrix. Each row corresponds to the first phase of labeling, and each column is the second phase of labeling of the same spectra. And you can see that it looks rather similar to what we saw in the model. Now, I must say that I used, in my model, I only used the spectra which are here on the main diagonal. So the spectra which were labeled the same in both iterations. And I will just let you compare. So the three tables that I already showed you, the F1 scores on the original data set, and the updated data set, which included more data, and then also a column for the rate of consistency. So take from that which you will. And I will end on some future considerations, because arguably there's a lot that can be done here. One idea would be to modify or replace the architecture or the hyperparameters of the network, because who's to say that this choice was the best one? It is always an option, but going deep into the hyperparameterization can lead you down to a rabbit hole, and then you don't know where to stop. Another idea would be to introduce some post-processing steps to refine the final labeling, using some localization constraints, possibly. And finally, to integrate this method into Spectra, which is software developed by Nuno, which is used to perform MRS analysis. And here I would thank you for your attention, and yeah, feel free to ask questions. Yes,
Thank you
please.
for the nice talk, and I'm seeing there is a question.
Yeah, I'd like to ask you, have you tried this on patient data? Something I worry is that you may reject spectra with lipids or only noise, because that's actually the characteristic of the lesion in the patient.
Yes.
And you would end up like throwing out that spectrum. So it would be a bad spectrum because of the pathology, but not for any technical reason.
Yes.
And so I worry about, using just, the presence of noise or lipids as exclusion criteria.
Yes. So just to clarify, when I talk about the four classes, I'm not saying that something is necessarily rejected immediately. I do argue that the useful or good quality spectra are easier for quantification purposes, but indeed, we can expect to see maybe the upper right class or the lower left class in a lesion or in a tumor. And it does show up, and it classifies it as is. But when you look at the results, when you overlay them up, it also shows it could help to use the decision of the model together with the qualitative analysis as such, and to see in which region you get classes that you would normally not expect there. That answers the question.
For the classification, did you use just like a normal multi-class classification, or you did?
Yes, so it's a very simple multi-class classifier with a categorical cross entropy as a loss function, but
something very easy to try would be just ordinal classification, where it gives a higher weighting for, if the model says a 0 is a 3, that's really bad. Whereas if the model says a 0 is a 1, it doesn't weight that as poorly. So that's just something maybe to try.
Yes, yeah. One of my ways to maybe compensate for that was to introduce this ensemble of classifiers, which would add more. Well, my argument is that if we have 3 classifiers, one of them says it's a 0 and 2 of them says it's a 3, that it adds a certain weight to the decision of the 2 that said 3. But indeed, that's a very nice suggestion. Thank you.
So due to the time restrictions, we will take one last question, and then thank you. Apologize for that.
We can talk.
You can ask your last question, Anke. Yeah.
Wanted
a quick comment to maybe back Peter with his comment about the noise and the lipid. I think in tumors that is highly relevant because it has a necrotic core where there's indeed noise, there's just water, and there's a lipid ring. But what you could do is adapt the criteria to the disease. So I think in tumors you have to maybe define your rejection criteria a little bit different, but in a lot of other diseases your criteria are perfectly fine.
Yes. Yeah, thank you. And I think that it also goes well with what I said in the end, which is that some additional post-processing steps can be included to add some fineness to the outcome of the model. So adding localization, for example, constraints would already be helpful in that sense.
I have a very burning question that cannot wait. If the panel allows me.
What can I do?
I want to ask you the opposite of, so when you remove these voxels, you end up sometimes with holes in the image. Do you have is there any consensus, or have you tried to look into methods to fill those voids? Like when you show those images with holes to radiologists, they may have some comments back to you in terms of whether they like it or not. So is there anything that you looked so far, and or what do you think is the best thing to do?
I'm not sure I understood. Could you maybe clarify? I'm sorry.
All right,
when you filter by quality voxels, you end up discarding some of your pixels from the image.
Yes.
Sometimes you remove an entire cluster of voxels, so you create a hole in your image.
Right.
So have you looked at ways to actually refill those voids, like not end up with an image which is maybe has empty holes into it, like Swiss cheese, but
right.
More
well,
like
it's not physically just taken out. It's assigned a label, so you know that it's maybe discarded, not worth quantifying as a spectrum, but it's still very much present. Yeah. Thank you. I'm sorry.
Thank you so much
Yes.
for this nice
Thank you.
talk and discussion.
Yes.
So the title of the next presentation is Effect of Circadian Rhythm on Brain, NAD, a magnetic resonance spectroscopy study at 7 Tesla, given by Ziwei Huang from École Polytechnique Fédérale Lausanne, Switzerland.
So hello, everyone. I'm Ziwei, and I'm a doctoral student at EPFL. I'm glad to have the chance today to present my work, Effects of the Circadian Rhythm on Brain NAD Level, an MRS study at 7T here. So first, as a brief introduction, the circadian rhythm, the circadian rhythm is a process that regulates our daily lives according to the environment, which repeats roughly every 24 hours. For example, we tend to feel sleepy at night, and we could more easily get focused and work high efficiently during certain periods in a day, like now. So with the discovery of the circadian clock, we know that these behaviors are partly encoded in our genes. And since then, our understanding, our interest towards the circadian rhythm has since expanded to explore the molecular basis of it in order to provide some insights to the physiology and diseases. Up to now, it was found that the NAD is directly linked with the circadian rhythm. NAD is an important metabolite involved in bioenergetics for brain metabolism and ATP production. It exists in an oxidized form as NAD+ and a reduced form as NADH. The redox ratio of NAD is also an important indicator for maintaining the cellular and mitochondrial hemostasis. Previously, it was found that the NAD+ oscillates around 30% in the mice liver, and NADH oscillates around 30% in human red blood cell during the day. Besides, lactate is another metabolite which is important in the brain. as it is the major energy source for the brain cells. One lactate is generated from the pyruvate with one NADH molecule being converted to one NAD plus molecule. A 24-hour rhythm in the somatosensory cortex of the lactate was already found in the rats, with a higher level of the lactate found during the active phase. Besides, us human also tend to exhibit a higher reward and risk propensity in the afternoon compared to that in the morning. However, all of the molecular findings shown here still stayed on the phase of animal experiments or just cell or tissue experiments, and it remains a challenge to translate these insights into the clinic. Thus, the goal of this study is to explore the molecular basis of circadian rhythm clinically. And the main hypothesis of this study is that the brain NAD level is modulated by the circadian rhythm, and we expected the NAD redox ratio to increase while the total NAD level not to change during the day. As a secondary endpoint, we expected the brain lactate level and the risk-taking willingness to be correlated with the circadian rhythm. Last, we are also interested to explore the possible associations among the measured metabolites and the behavior test results, if exists. To do this, we recruited 25 male subjects in order to be able to catch 10% of the change in NAD level if exists. And our experiments were conducted at 2 different time points during the day, one in the morning at 8 a.m. and one in the afternoon at 3 p.m. So during the experiment, the diet was controlled and 1 week sleeping diary prior to the experiment were recorded in order not to have anything interfere the rhythm. Basically, the protocol for both sessions are the same. So for each session, we first gathered the salivate of the volunteer in order to measure the cortisol level. Then MRS scanning was implemented in the occipital lobe. A pulse acquired sequence with a phosphorus coil and a STEAM sequence with short echo time were implemented to measure the phosphorus metabolite and proton metabolite concentration. At last, the volunteers were asked to finish a balloon analog risk-taking test, namely BART test, to measure their risk-taking willingness. After the experiment, the spectra were processed with MATLAB. So first, phase and frequency correction were implemented to both spectra, and eddy current correction were done to the proton spectra. After this, the scans with low SNR were discarded. And after the whole experiments were finished, we performed paired t-test in terms of all the measured metabolites to compare their difference between the morning and the afternoon indices. And here shows the sample spectra. For the proton spectra, 20 metabolites with CRLB lower than 20% were kept for further analysis. As we could see here, the spectra are in general of good quality. And this is the main peak that we would like to measure with the proton MRS. The CRLB of the lactate is around 6%. For the phosphorus spectra, 11 metabolites with CRLB lower than 20% and NADH with CRLB lower than 40% were kept. And these are the small peaks of the NADH and NAD plus that we would like to measure. So according to the statistical analysis, we found that the cortisol level was significantly higher in the morning compared to that in the afternoon. This confirmed that our experiments were actually implemented under 2 different circadian status. No significant difference was found in terms of the NAD level and the NAD ratio. However, we found that the NAD ratio has much higher variance in the morning compared to that in the afternoon. Besides, we besides, we found that the BART performance is much higher in the morning, and the lactate level is positively correlated with the NAD+ concentration. Based on the previous findings, we divided all the subjects into two groups, the low group and the high group, relative to the median value of the NAD ratio in the morning. And then we implemented paired t-test again within each group. And this time we found that in the low group, the NAD ratio has significantly increased in the afternoon, which is mainly driven by the decrease of NADH. In the high group, however, the exact opposite was found. Based on this, we inferred that there might be two different metabolic pathways going on. Yeah, and here shows the p-value. So to sum up, we found that those with low NAD ratio in the morning have much higher NAD ratio in the afternoon, driven by a decrease of the NADH, while the high group is the opposite. This indicated that there could be two different metabolic processes. Besides, we verified the previous assumption that people tend to exhibit a higher risk preference in the afternoon than in the morning. So all in all, this is the first study investigating the effect of circadian rhythm on brain NAD in humans. And I would like to thank Kazia for helping using the centrifuge, and Mark, Songyi, and In for developing the sequence. That's it. Thank you.
Thank you. Any questions? Here.
Thank you for the presentation. I would like to ask whether have you measured individual chronotypes of your participants, and do you know how did influence your results, whether there are some
What?
individual chronotypes? I mean, whether the participants are more active early in the morning or
so we have the so prior to the recruitment, we have all the volunteers filling a form called the morningness, eveningness form, and we only selected those are not extreme morning person and not extreme evening person.
I'm not sure that if there's an easy answer, but I would assume that the NAD total NAD plus NADH value should be stable, highly regulated. Is it something that you could use in your analysis or that you have observed? I'm not sure about the results at some point, because there was so many graphs.
So the total NAD level actually stays stable across the day, if that is your question.
Yeah, that was mostly the question. And you observed that, right?
Yeah.
Okay, thank you.
So I had a couple of questions about sort of the study design.
The first is it true that all of your subjects, all of the PM scans were second scans, so there's a potential scan order compound between morning and afternoon?
It's the same scan in the afternoon, yes.
But you didn't have any afternoon subjects who hadn't had a prior morning scan?
So all the subjects did the same two tests, one in the morning and one in the afternoon.
So you don't know whether you're studying circadian differences or scan 1, scan 2 differences?
I think so the scan results are quite stable. So yes, we're studying the scan 1, scan 2 difference. We could say that, I guess.
The other concern I have around is when you're stratifying based on the NAD ratio and then showing statistical differences on sort of non-independent measures, that has all kinds of statistical concerns.
In what sense?
Well, you're sort of you split into two groups, and you say the low group goes one way and the high group goes the other way, and this sort of feels a bit like mean reversing.
Oh, okay. I will think about this later. Thank you.
More questions?
Hey, thank you very much for the presentation, and sorry if I missed this information, but why did you include only male subjects? Is there any-
Yeah, because a previous study showed that male and female have different hormonal level, and we don't want that to interfere with the study.
Okay, thank you.
Just a comment, a follow-up comment to the previous questions. This sounds like a classic regression to the mean. The people who start out low go higher, and the people who started out high go lower. Just a consideration for you and your team.
Yeah, I understand. To me, this kind of makes sense now, but I will think about it in more detail later.
I think there's no further questions, or maybe one more. Yeah, two more. Okay.
Very nice talk. Thank you. I wonder if you were able to get an independent subjective evaluation of people's risk-taking propensities to compare with the possible two groups.
So sorry, I didn't hear it.
Did you get people's own subjective ratings of how high levels of risk-taking they saw themselves?
You mean how we evaluated that, or?
Whether you did evaluate that?
So we used the BART test, and we just adopted the indices proposed in that paper, and the index is the average pump that the average pumps that they clicked before the balloon explode, and we simply compared that value. If this is your question. Sorry.
Did that correlate with the grouping that you saw?
We didn't compare that.
Okay, one last question, I guess.
So if my understanding is correct, you're looking for a circadian rhythm, like, and you have 2 time points.
2 templates?
Am I wrong? Did I miss that?
What do you mean by templates?
Time points.
Yeah, time points. Okay.
Yeah. So how do you know that, and maybe this is like another way of saying what 2 other people have commented, but it seems to me it's, how do you know that you didn't miss the peak or the trough of their rhythm with only 2 time points?
Yeah, I think this is a more or less a trade-off problem. So we tried to so for the morning session, we tried to do the experiment as early as we could, and for the afternoon session, we just select like 3 p.m. so that, because we cannot do it like for 3 times until the evening, I don't think the volunteer will be happy with that. So this is just like a trade-off.
Okay, I think there is no more questions. That will close this session. Thank you so much for your interest.
Okay, thank you. Thank you.
Okay, so welcome to the last and final session of today, which is about accelerated MRSI and reconstruction. So this is my co-moderator, Antonia Schuschnia, and I'm Bernhard Strasser. And so we will start with the first presentation by Ovidio Antonesi from the Martino Center. And sorry, I have to say, like, the third presenter, Jack Heighton, didn't upload the presentation. If you're here, maybe can you please upload it now? Is Jack Heighton here? Okay, so maybe we'll have to skip this talk. Let's see.
So should I start?
You can start. Yeah, thank you.
Okay, welcome everybody back. I know it's very hard to compete with the nice weather outside, and thank you, Christina, for organizing this workshop here in this beautiful location. I don't know how you guys work here when it's such a nice weather. I think I'll be able to work only when it's raining, like before the weekend. I'll try to keep you inside if I can with this presentation and try to finish in time so that we can go to what was planned after. So you probably heard already about many of these things that are listed regarding the value of MRS and MRSI. We can compare it also to the other molecular imaging, for example, PET or SPECT, that we have advantages in terms of value also compared to the other molecular imaging approaches. However, we also have problems together with these advantages, in particular for clinical protocols. So one of the problems is that it's slow, more than 10 minutes, or depending on the resolution that you want to achieve and so on. Besides being slow, is also low resolution, so slower, lower than the other molecular imaging, so 10 millimeter, or it can be even worse. And we don't cover, with some of the clinical protocols, we don't cover the entire brain, we have partial brain coverage. And these three things are related to the fact that for the long acquisition time, we need actually to do a 4D KT space encoding. So we need one extra dimension that needs to be encoded, so that requires time. The low spatial resolution is due to the fact that the concentration of the metabolite is much lower than water that is used in MRI. And partial brain coverage is one of the solutions to cope with these lipid artifacts for proton MRSI, but also B0 inhomogeneity. And so we have this, we could say we have this situation that we have a high value, but relatively low performance and also low quality in the same time for some of the clinical protocols. And because of that, we have to deal with this fame also for MRSI on the MRI Hall of Fame, especially if you go to radiology meetings and other meetings that are more clinical oriented, you will hear the comments like this, that MRSI has been the next promising imaging to try for the past 40 years, but also from more skeptical people is that, and likely will stay like this for the next 40 years. So my proposal for you with these MRS workshops would be to, and the work that we do is that maybe we can set to shorten this interval till to be listed properly on this MR Hall of Fame to try to do as much as we can and shorten this to an acceptable time for our careers, so before we retire. If possible, in the next 10, I don't know what I'll do past 10 years, but within the next 10 years, probably I'll still be active, okay, in this area. So now I'll do a comparison between the, so to give you an understanding why it's slow, why we have low resolution, and I'll compare what how data are acquired in MRI and how data are acquired for MRSI, and how we can accelerate that. So, and I'll use as a toy example the simplest sequence that you may imagine for MRI. This is a 2D slice encoding sequence, and you see that this sequence uses the three types of localization that most of the sequences use in MRI. So slice selection, phase encoding, and frequency encoding. So almost all the sequences use this. There is an additional dimension when you have multiple coils, which is the coil, the localization given by the phase arrays or spatial gradient coils. And so you start with a RF pulse that excites your spins in a slice by simultaneously applying an RF pulse that has the bandwidth matched to the gradient amplitude used for the slice selection, and this you select along the Z direction. And to localize your image along the other two, Y and X, you apply frequency coding for X, and in successive TRs, a so-called phase encoding. And if you do this, you acquire the data that you acquire is not in the image space, it's in the so-called K space, the inverse space of the Fourier inverse space of the image space that we are used to look at. And this sequence acquires this space in line by line. Yeah, so it's relatively fast, specifically if you have very short echo time, very short repetition times. Like if you have TRs of a couple of milliseconds, this can be very, very fast, even for a high matrix size. Now, if you use the same sequence for MRSI, there are two major changes that have been done, and some in the previous one of the previous presentations that has been already alluded that. So because of so in spectroscopy, we are interested now to identify multiple frequencies, multiple peaks. So in MRI, you have only one frequency of the water, and that's all you care. But in spectroscopy, we have peaks at 2 ppm, at 4 ppm, and so on. And to be able to distinguish all those different frequencies, our readout or this FID that you see here has to be much longer than in MRI, so on the order of a couple of hundreds of milliseconds. So typically you'd have a TR of at least 200 milliseconds, but way longer in many clinical papers that you read. So you cannot shorten this too much, because then all your peaks will be on top of each other, or you have ringing artifacts and you cannot separate them so well. And because you need to separate those peaks, people have removed the frequency encoding gradient, because that gradient smears out all of these frequencies and broadens them beyond the recognition. So you don't, then you lose the ability to identify these different peaks. So that's replaced the frequency encoding. So the first two things stay the same, the slice selection and the first phase encoding step. But the third step along X, which was frequency encoding and was very fast, so you acquire in a single TR an entire line in K-space, now becomes a phase encoding, which means that you can acquire only 1 point per TR with the sequence. So simultaneously, you acquire 1 point per every TR, and also this time domain, this FID. But this is very slow for the spatial encoding of the K-space. So line by line, if you have a TR of more than 200 milliseconds, that this time easily goes into hours. If you go to matrix size that are beyond 64 by 64, and if you add also a slice like a Z direction, yeah, it's something that's not feasible. Yeah, so clinically, but even for working on healthy subjects in neuroscience type of projects, that's not something that, because of many reasons, motion, scanner drift, and so on. So yeah, just to make this more clear, I have this animation. So we see, so for a 3D MRSI, we have a 4-dimensional space that we have to acquire. So there are 3 spatial dimensions in K-space and 1 time dimension along the FID. And basically what we do for every time point along the FID, we acquire this K-space again and again and again. Yeah, so we take 1 time point, we acquire 1 full K-space, we move to the next time point, we acquire again the same K-space, and so on, and so we get to this 4. This is very similar to fMRI, for example. People that do fMRI, this is a dynamic scan, but only that our repetition frame is much shorter. It's not on the order of seconds, it's on the order of milliseconds that we need to fulfill. And with 4D Fourier transformation, you go from this space that you don't understand. When you look at it, you see something like that. So typically, there are 2 ways to look at this: the MRSI data, either spectra on top of some anatomical image. This is probably, I mean, it's still used and we are still using it, but it's yeah, requires more time to pick through all of these different spectra. We can produce metabolic images from all of these peaks that we can identify and fit, and we can obtain, let's say, if we can fit 19 metabolites in the spectra, we'll get 19 metabolic images at the end, which is pretty awesome, because there is very few other metabolic images that we would be able to get 20 different metabolic images simultaneously with no other. So for example, if you compare with PET, imagine that you have to image 20 PET tracers in the same subject to get all of those 20 different images or for specs. So it has high value, but as most of us that work daily with this, we still deal with low performance and low quality. Okay, and this has been already mentioned, these FAT artifacts. This is another way to maybe looking at this data. You'd probably understand even maybe visually makes it a little bit even more obvious what you are dealing with. So the fat comes only from the scalp around the brain in this healthy subject. But because we have low resolution, so a point spread functions with a lot of side lobes, that high and a very high fat signal, which is about 100 times to 1,000 times higher than metabolites, that ripples from the side lobes are so high that it dominates the metabolite because of this 100 times factor. So if you look at the blue spectra, which are before you do any fat removal, anything with the fat, you mostly see a lot of fat, and the metabolites, you have a difficult time to feed them. If you remove them, suppress them with inversion recovery, sidebands, if you do some post-processing fat removal, then you get this green spectra, where you can do a better quantification on what you have there. Yeah, and if you look at the maps, so the upper map is what you would get if you don't care about the lipids and you don't do anything. So like an NAA map, where you see a lot of waves and a lot of, it looks more like, I don't know, a supernova or some a black hole or whatever. And if you clean this, these large lipids artifacts, you can get a more a better looking but also more exact, more correct image for this metabolite NA. Okay, so some of these methods have been mentioned. I don't go through them. So you basically acquisition or processing, and the conventional ones that are used are either to avoid the scalp by doing a volume of interest excitation first. So you do a press, steam, or laser, or spatial, or other types of localization of a big cube inside a big box inside the brain, but that's which avoids the scalp, but also avoids some of the periphery of the brain. You can place these saturation sidebands they also cannot be placed really perfectly only on the scalp. You cut out through the brain, so you miss cortical regions. You end up with octagonal looking brains instead of nice elliptical brains with this OVS and so on. And you can do post-processing with several methods. One of them that performs quite well is this lipid subtraction based on the difference in spectral and spatial support, or like orthogonals and lipids, and which has been used for the data that I showed you before to be cleaned for metabolites. Each of them has several pros and cons. And to move to the real part of my presentation, the need for optimized MRSI acquisition is that why do we need this? Is to probe brain compartments that have complex shapes, visualize small lesions, metabolic heterogeneities, boundaries of large lesion to better address the demand of clinical users. We need to be faster to be clinically acceptable, and also be subjects to be more compliant with this. We can reduce artifacts due to motion because there is less likelihood for motion or scanner instability, and whole brain because we don't need to miss anything that is important. Okay, and you can see examples here in the literature where, for example, you can see this is a demonstration data that demonstrates metabolic heterogeneity in some of the metabolite maps. For example, this is 2AG in glioma, and actually you have different heterogeneity between 2HG and choline. Or in this other study, this was done in multiple sclerosis, you see the effect of going from high resolution to low resolution. So compared to the flare, for example, here you see the hyperintense lesion of multiple sclerosis on flare. You can see them on this metabolic map from MRSI at high resolution, which is close to the flare. You can still see most of them when you go to 3.4, but you lose a lot of them when you go to 32 by 32, which is pretty high compared to clinical images. But in this, or clinical MRSI protocol, but in this case was the lowest resolution that was employed. And this is because the metabolism of the lesion is diluted by the healthy brain around it. So how can we accelerate? So there are two ways basically in MR. So either we acquire more case space per slice in a single TR, either with gradients, this spectrospatial encoding with SMS, SPAN, skip case space or time domain samples with parallel imaging, compressed sensing, SPICE, Obviously, you can combine one or two, and the simplest is to shorten the TR, but you cannot do it beyond a certain point. This is a nice review paper that goes, so I'll mostly focus on this spatial spectral encoding. And the next speakers will speak about the combinations and more fancy and more advanced methods that I have time to go through. So here we play, and I'll contradict a little bit the previous speaker or also myself at the introduction. We actually can use readout during the FID, also for MRSI. So these are some trajectories that you can play during FID readouts, spirals, EPSI. Some of them have been introduced in the 90s. More recently, concentric circles, radials, rosette, particularly the circle trajectories. And another trajectory that uses circles, so-called eccentric. Then Antoine, which is speaking after me, will give you a more details and more advanced presentation that I'm able to do. Or so these are randomly placed circles in the K-space. These are in a parallel fashion wave Kuiper circles placed. And so what you can see here is that a lot of trajectories use these circles placed in a certain way. And the reason is that to produce circles trajectories, the gradients, you play the same gradient all over during the FID with no change in slew rate on the amplitude. This is relatively easy for the gradient hardware and so on. And so which of these is best? So, and now the answer is also in that review paper, so I'll advise you to go into it and read it. So the answer is the one that we love the most. It depends. So it depends what you want to do. So if you want to do high field or low field, 7 Tesla or beyond, or 3 Tesla and below, if it's X nuclei or proton nuclei, if you want speed, but most of it you want it for speed, or you want high SNR. And they have done an analysis of how speed changes with resolution for different trajectories, going from spiral to concentric rings and EPSI, how the SNR efficiency changes again with resolution and or the bandwidth. And so I don't have time to go into all of this, but that's a good comparison that's in this review paper. One of the most, let's say, solid packages that is out there is based on EPSI, which has been, and that's the work done in the group of Andrew Mosley. He had a long time to develop this, and you can see like this is probably one of the still some of the best maps that you can see published in regarding MRSI, where you can see water from MRSI data, unsuppressed MRSI data, then metabolite, then quality maps at the end below. I'll go in the next, I'm a little bit almost over time, on how we can actually play this gradient during the FID readout. So, and I will exemplify this for spiral because that we have used the most. So this is the grid of K-space data that we want to acquire, and the Cartesian grid is shown with the black crosses and the spirals in blue and with the spiral equations. And what we do is in a single TR, we acquire all these blue dots that are on a spiral by playing these gradient waveforms. Basically, this is a circle of increasing radius. That's what a spiral is. And you can see it in the gradient waveform. It's a sign that has the pitch is changing, but also the amplitude. That's how you get the spiral. And how can you do that? Well, for MRI, they can basically play a spiral from all the way from the beginning of the readout till the end, one single spiral. If we do that, we run into the problem that we smear the peaks and we don't see them. So we have to make a small change. We need to play one spiral per single FID point. So for every FID time point, we play one spiral, and the duration of the spiral is determined by the inverse of the spectral window. In that case, we simultaneously do this K-space readout in a single TR plus the time dimension in a single TR. And that's how we can do. But we have a serious limitation. We cannot travel too far in the K-space in a couple of milliseconds, which is the duration of this time. So we need to play interleaves images, but so angular interleaves and temporal interleaves. But the time savings are very big. So like for a small matrix of 16 by 16 by 8, that takes half an hour with phase encoding, now it's under a minute. And this is the acceleration factors, about 40 to 50, you can get, these are times at 3 Tesla, and you can see the improvements in point spread functions that going from the highest resolution in red to the lowest in blue, in black here for phase encoding. One word that I can only say for the now we put the emphasis on the reconstruction more than the acquisition. For all of these advanced methods, you have to have more fancy reconstruction than for simple phase encoding. The good news is that a lot of this has been worked out by the smart people in the MRI community, that they have implemented first for MRIs, spiral, CPI, low rank compressed sensing, so we can piggyback on what they have done. We need to do some changing adaptation. It's a little bit more trickier for us because we work in a low SNR regime, very sensitive to motion compared to them, but we can build on what is already has done by MRI and try to adapt for our own use. So for non-Cartesian spectral spatial encoding, there are basically two ways to reconstruct your data. Either you grid the non-Cartesian data first to Cartesian case space using a Kaiser-Bessel window function, and then you do the normal Fourier transform, or you do the non-uniform discrete Fourier transform. And that's what's behind all of these maps that are produced with spirals or circular trajectories. Now, towards the ultimate MRSI is, so also MRSI is not as complex as the equation of everything. I don't know, like for those of you that are here by training physicists and have studied particle physics, you easily can recognize this nice formula here, which is the Lagrangian of the standard model for the particle physics. You see a lot of terms here, like with MRSI recon, we are not yet to the same number of terms, but we are starting to approach like with some of these reconstruction methods. So maybe one possible challenge for the next MRSI is let's go beyond the standard models in number of terms, not just for the sake of it, but just for to trying to improve what can be done. And this is a demonstration of what, for example, what high resolution can buy you with these parallel images. And you see, for example, this in a glioma patient, phase encoding data at 1 cubic centimeter, you see some kind of a blob where a tumor is of high choline. At the same resolution in spiral, which is 4 times faster, you get similar blobograms. If you improve the resolution, let's say 0.4 cubic centimeters, but in half of the time you start to see some spatial features here that are resolved, and which actually make more sense when you compare it with the contrast enhanced T1 image that you see here on the right side. Okay, so I'll go over this because this has been covered. And so you see data here produced with concentric rings at 7 Tesla. They can do close to 3 millimeter or even under 3 millimeter. And depending on the implementation is on the order of 15 minutes, give or take. And with undersample, as I mentioned, this can be combined with parallel imaging undersampling or compressence. And this has been already demonstrated for MRSI. This is just parallel imaging alone. This is compressing alone, and this is combination of spatial spectral encoding together with either parallel imaging or compressing. And so to summarize, these are my 3 last cows here listed. So with fast high resolution whole brain MRSI is possible with spatial spectral encoding. So it is possible to accelerate between 50 and even 100 times acceleration times at 3 Tesla compared to phase encoding. And note that when you do parallel imaging or compressed sensing, your acceleration time usually is less than 10. At 7 Tesla, this acceleration decreases because we need to fulfill the spectral window dimension. So, but we still can do probably better than 10. We can probably approach 5 millimeter in plane resolution at 3 Tesla, 3 millimeter in plane resolution at 7 Tesla, with 10 millimeter slice thickness for full brain coverage.
And with acquisition time that ranges anywhere between 10 and 30 minutes, but that depends also on the TR that you use. If we combine it, if we can, so this is spectral spatial encoding alone. If we combine it with the undersample key space acquisition, parallel imaging, and compressed sensing, then we can push it everything a little bit, the resolution, but also the time until it breaks down. And then, but we also require robust methods for high spectral quality for clinical use, and that includes B0 shimming, dealing with lipid artifacts, at least for proton MRSI and motion. And I'll point you to these review papers that have been published in this special NMR biomedicine, but also outside of it, radiology or MRM. And I would like to thank you for listening and acknowledge all my the people that I have worked before, and they are brilliant in all their work, and both from MGH, but also outside of MGH, and I'm well over time.
Thank you. Is there any quick question? If not, maybe in the interest, oh yes, there is, please.
Comment on the question. I'm not sure you understood the rules with cowhead. I'm just wondering about
I have also hidden cows. Like those are the cows that were not hidden. The last three ones. Yeah.
When you're in a circle or a spiral in phase space, I understand that's easy on the gradients when you're close to the center of phase space, but when you're trying to capture the edges of phase space, isn't that very demanding on the gradients also?
You go to, yeah, I mean, with spirals, the gradient amplitude increases as you approach the K space. So yeah, you reach a limit in the K space. With the circles, especially if with the eccentric or the wave, no, because you play always the same circle, and you move it around. So you always have the same gradient readout. It's a constant radius of the circle, and you can choose your radius in such a way that is not demanding for your gradient. It's not a center. You choose, let's say, the way Antoine will have more time to explain you, but let's say you can use the circle of the radius to be 1/5 of your K space max, or 1/4. And that is true for all the circles that you play, and you move that circle around, and you don't exceed that.
So thank you again.
Yes.
Before we go to the next speaker, is the Jack Hyten somewhere here around? Yeah. You didn't upload your presentation. Can you do that maybe now or during the Q&A of the next, like right before your talk? Okay. Yeah. Okay. So the next talk will be given by Antoine Clauser. I'm from the CIBM Geneva, and it will be about advanced techniques in MRSI.
Okay, sounds good. Okay, good afternoon, everyone. Thank you for to the organizer to giving me the opportunity to give this talk about the advanced techniques, and also thanks Ovidiu for the nice first talk. Actually, I'm gonna build up on a few of these concepts to go even further in acceleration, or to go more into the detail on how we combine the different ideas that have been presented. So during this talk, I'm gonna go through first advanced techniques, a bit why do we need them and how we're gonna implement them. Then the key concept I'm going to talk about in this talk is the inverse problem solution. It's going to be really a general concept that we can find in different application, different encoded scheme, and that's really the basis for high acceleration. And eventually, I'm going to also talk about the denoising that will be necessary. So advanced techniques with accelerated MRSI, it's a bit, can be a lot. I could talk a lot about different things, so I had to choose. So as I just said, I'm going to focus on the reconstruction scheme using the inverse problem. It's going to be really the main idea. I'm going to talk about, we can also add some denoising on top of that, and we're going to see a few examples. I'm not going to talk about specific excitation or specific magnetization preparation. Basically, they are not specific to MRSI, and neither about super resolution nor AI reconstruction, although very interesting. I mean, I just had enough material with what I wanted to present. So why? I mean, looking at the stamp of the MRS workshop 2022, it says overcoming the barriers to clinical use. So why coming up with more advanced, technical, difficult to implement methods? So Ovidio already presented some advantages, but of course now the goal would be to acquire the whole brain in high resolution, a bit like a new imaging modality. So we can determine the viability pathology of a specific lesion in the surrounding without having to position any specific region of interest. The region of the analysis could be done a posteriori. And of course, there is always a contralateral comparison, of course. And also the placement of a high resolution whole brain is quite easy. When I'm talking with technicians, sometimes putting a single voxel on a specific structure, they're not really at their ease to that, but just positioning such a volume field of view, sorry, it's much easier. So starting from this kind of protocol, 5 mm isotropic, FID MRSI, short TR, phase encoded acquisition, simple calculation leads us to a 2-hour acquisition time. So again, as Ovidio introduced, there is a need for acceleration in order to reach an acquisition time that is valid for, that is useful for clinics or research. We should go to the order of 10 minutes. Then, of course, reducing the voxel size to 5 mm isotropic or even lower, we'll need some denoising methods to compensate the drop in SNR. And of course, we have to cope with the lipid contamination from the skull. That's the last element. Ovidio again talked a bit about that, but that's good because then I can go quite fast on that. But we've got a problem. Now we got an excitation that is not selective, middle of the brain. We excite the entire slab. There is some lipids by the skull. They are excited, they produce some signal, and they will contaminate actually the whole field of view through the Pons-Frouet function. Only due to the difference in order of magnitude between the metabolite signal and the lipid signal, they will just go contaminate the whole field of view. You can see here this example where you can see a box in the middle of the brain for a 2D FID MRSI acquisition at 70, and lipid contamination, the presence of lipid is too high to make it any kind of quantification possible in this case. So there is a real need to use lipid suppression technique. Could be different type. Also refer to the nice review paper and this nice paper by Engel et al. Where they actually review different technique for lipid suppression. Could be RF pulse for inversion recovery, double inversion recovery, case-based filtering, extrapolation, or model-based removal like the L2 norm reconstruction methods. But from now on, I will not talk about this lipid problem and suppose that lipids are completely suppressed from our data sets. I'm only going to focus about the reconstruction of the metabolites. Although this is far from a solved problem. I mean, there are many methods and they all have the drawback, but still, I mean, that's probably a subject for another talk. So the reconstruction using an inverse problem solution. Introducing the inverse problem. Inverse problem is something that happens in many different fields. Of course, we talk about MRI here, but it's used in microscopy, it's used in astronomy. As soon as there is something in a real world we want to look at, we want to record, and we use an apparatus for that, and we record some raw data, might be an inverse problem solution. So in our case, we are interested in magnetization or distribution of metabolites in the brain, and we measure it with an MRI, with a Ford model. That's a physical model that includes the encoding with the gradient, the playing out of the gradient, maybe the RF pulse or the B0, B1 inhomogeneities. And eventually we record some raw data in K-space, or could be more complex for non-Cartesian data. The forward model is well known. It's generally well known, and it's very easy to describe the let's say the forward direction. How from the real world we're going to measure and produce some data. And generally it could be expressed in a single equation like this one, where we got our real world distribution of metabolites. We got the forward model, it's a linear operator, and we got our measured data plus some noise, Gaussian noise. And for instance, the forward model could include a spatial encoding, cold sensitivity profile, of course, if we measured with a call with many elements and some B0 inhomogeneity. But that's an example. It could be made of different elements and different operators. The inverse problem comes from the fact when, most of the case we are interested is that we acquired some data and we want to reconstruct the magnetization in a real world. And this might be a problem because it's often ill-posed in a sense that as soon as there is some acceleration or there are some point missing in a K-space, there is not only one solution for one set of raw data that are acquired, ill-posed problem or underdetermined problem. So there is no easy step how to reconstruct the data in an image space. And most of the time, at least most of the time, the problem is solved by iteration using an optimization, minimization approach as presented here. And that's the way you're going to also find it maybe in different paper explaining different approach in acceleration. And this line here means that we want to minimize a cost function that is introduced here, and we will find the solution rho. That's going to be our solution that minimizes this cost function. I am a physicist, and that was some signal processing knowledge that I got to use, but I see that as an energy problem. And studying the dynamics of a system, often we know that the steady state of a system is at the minimum of the energy. That's a bit the same here, where the proper solution would be located at the minimum of the cost function, of the minimum of the energy. And so this cost energy is going to be described by data fidelity terms that actually measured how far our solution is from the measured data, and we want to keep that as small as possible, and some regularization that is some prior knowledge. I will get back to that. And eventually, this problem, again, cannot be solved in one step with a single linear operation, but we need to do it iteratively using a gradient descent, basically taking the gradient of the cost function. We'll start at an initial condition and following the gradient to go to the solution minimizing the cost function. Here to help visualize that, we put a 2-dimension, but of course here is a space of all possible solution. So rho being our MRSI data sets, a 4D data set. So of course it's much bigger than how it looks here, but it's good for visualization. And so doing this reconstruction by iteration, there is a it's very important to choose the right regularization, because this is the term that's going to help to solve the ill-posed problem. Using only the data fidelity, we are still stuck with the same problem, meaning that we got some raw data and it exists many solution in the image space, and that's only the regularization that's going to tell us this is the good solution. This one only is going to be the one to pick. And for instance, with compressed sensing, the regularization is a wavelet L1 norm and the finite difference or the total variation. And it means that the solution will need to minimize these two norms. And basically, it is some prior knowledge that we put in a system, but it is based also on the knowledge that we know from natural images. And we know that they are supposed to minimize these two norms. So that's the way that's basically the way we solve the ill-posed problem. As a first example, I would like to highlight the result by Nasirpour et al. That implemented compressed sensing for 2D FID MRSI at 9.4 Tesla. You see also from the manuscript, a very similar expression of the cost function. Argument to the minimum of the x is gonna be the solution. So we minimize this cost function. And again, in this cost function, we've got something very similar to compressed sensing, in a sense that there's a wavelet norm, L1 norm of the wavelet coefficient, and in total variation, there is a minimization of the total variation. But here you can see, here the author also adapted compressed sensing to MRSI. Compressed sensing, of course, was developed for MRI, but here needing to adapt that to MRSI, the authors included also a regularization over the time dimension, and the total variation was minimized also along the time dimension. And with this reconstruction framework, they could demonstrate that it's possible to acquire the 2D FID MRSI 9.4 Tesla at with an acceleration factor of between 4 and 5. It's already a good start, but this is not enough. I mean, for 2D, of course, it's far from enough. We go to an acquisition time of 3 minutes, but when we have to acquire the whole 3D space, it's still that would be still not enough. And that comes also from the fact that Cartesian phase encoded MRSI is by nature slow. As Ovidiu introduced, we acquired as well one single point in a case space per TR with phase encoded MRSI. And we need to go faster than that. And for that, we need to use a spatial spectral encoding. So the trajectories, that brings an acceleration factor at least of 20. And that could be performed either in a Cartesian, spiral, or concentric, or rosette way. And this way, in one single TR, we acquire a full line. And combining this with some undersampling, we can really accelerate at full speed a data set. Another example here by Saucedo et al., where they look at the acceleration performance for radial echoplanar spectroscopic imaging, or EPSI, and they undersample that prospectively, this sampling. We find again a description of the optimization problem with a data fidelity term here, the forward model here, the raw data D here, and some regularization that is the total variation of the solution. Now, there is nothing that has changed since phase encoded MRSI, because the whole thing about the trajectory and the encoding is hidden in the forward model. That's the physical model that tells us how things are recorded. But basically, the whole processing of converging and doing by iteration, following the gradient descent, is the same. And they could demonstrate that data could be acquired by a factor 2 or 3, going from an acquisition with a 2D simulator at 3T from 6 minutes to 3 minutes, basically. Okay, you might say 2 or 3, that's less than what we have just seen with the Cartesian, but don't forget that now these accelerations add up. We have the acceleration of the echoplanar spectroscopic imaging plus this factor 2 and also the short TR. So now we are probably at an acceleration factor close to 50. If you are interested in this approach, also just picked up this poster by Bernard. Sorry, went a bit quickly. It's gonna show some results using a compressed sensing and concentric ring trajectory. So now about denoising, I explain how we can go fast, combining undersampling, inverse problem solution, and spatial spectral encoding. But now we have a resolution that's quite high and voxel size that is very, very low. And of course, we lose in SNR as well. And we are still interested in looking at something that makes sense. So what are the solutions for denoising? I'm gonna introduce here two different solutions. The first one is a low rank approximation that is widely used, or sometimes we talk about partial separability. And I'm gonna give you an intuition here about how it works. So the low rank approximation is the approximation that says that the MRSI data, characterized here again by rho, and depending on the spatial coordinate and time coordinate, could be frequency as well, could be expressed as a sum of spatial component, time, temporal, or spectral component. And, or with the illustration, Actually, I'm really sorry, but you don't see my pointer, or is that? Okay. Should I go? Yeah, but then I don't see. Okay. Doesn't mind. I will go on. On the right, you see the illustration of it. Doesn't work like that. And I'm sorry. Yeah, okay, I see now. Laser pointer. Yeah. Okay, I hope it's not too late. Not well. Okay. Going back to this low rank approximation, if we got our MRSI data set here, visualized on the right, every point in space is a spectrum here, or it could be a time series, and we approximate this whole MRSI data set by a sum of map time spectrum plus another map time, another spectrum, etc., etc. Most of the time they are ordered by importance, and we go from the most important map to the least important. And doing a low rank approximation will keep the, let's say, first K component of this decomposition. It's also quite intuitive why noise would be quite difficult to, I mean, it's an efficient way to denoise the signals, because noise is random. So it's basically a noise spectrum would be completely different from one voxel to another. So it would be very difficult to represent that as a map time a spectrum. So basically, that's a very, in a short, that's a very efficient way to denoise the data by keeping only the first K component to the reconstructed data set. And in this case, the cost function would be just rewritten, replacing the rho by the U times V, the set of spatial component and temporal component. And the minimization process now doesn't aim to find a solution rho, but a solution for U and V. That's also a big advantage because we have a much reduced degrees of freedom for the problem. So we should be more stable and faster. The second way to denoise the data is simply the regularization. This prior that we were adding to the cost function actually help denoising as well. Here is some example of some unpublished data on the FID MRSI at 3T acquired in 2D. The first line is the reconstruction without regularization, and the more you go down, the more regularization is added to the reconstruction. And at some point, we really see an improvement in the map that could be also visible in a spectra. Slight improvement in SNR, spectra are keeping their features, but we see a drop in a noise that could be measured also over the whole brain. There is a gain in 50% SNR and a drop in Kramer-Rao bounds, an estimation of the metabolite for the 3 voxel here. And this follows the same increase in regularization that's shown here. So with these two ingredients, the low rank approximation and the regularization, this is the key ingredient for denoising and to really reach high resolution MRSI in combination with the acceleration. Of course, now I have to speak about SPICE and spectroscopic imaging by exploiting spatio-spectral correlation. This was really a pioneer approach using the low rank and acceleration started most 10 years ago. And I'm gonna describe here the basic concept that was really the beginning concept. It has been a lot of development that they put with many publications, but then I'm gonna talk about the main concept here. This is an acquisition in 2 steps. First, there is an acquisition of the center of the K-space with a full spatial and temporal sampling, either with phase encoded or EPSI. And then it's followed by a highly undersampled acquisition, again using EPSI, going very fast and covering a large K-space to go to very high spatial resolution, but with a low bandwidth and a really low spectral resolution. Now, the key thing is to combine these 2 data sets together using the reconstruction model, again, a solution to the inverse problem, with now data fidelity terms when we see the 2 different components, phi, I think, being the temporal component, C being the spatial temporal spatial component for macromolecules and metabolites in this case, and with some regularization that have been chosen here, and D being basically the total variation. With this, they could perform 3D, almost a bit less than 3 millimeter isotropic in 5 minutes at 3 Tesla. And again, this is just the basic idea again of the, I think one of the first publication, because since then they improve it with using some more fancy undersampling scheme, some replacement actually for the first acquisition, they replace it by some AI and also having different type of reconstruction model that have been presented. But still, this is a demonstration of how to use a low rank regularized reconstruction in order to reconstruct a high resolution. Another example that might be a bit more at my ease is to explain eccentric. This is some specific encoding of the K-space combined with a low rank approximation. You might know it or not, at 70, when we want to scan at a very high resolution, it's difficult to perform. It's difficult for the gradient system because of the large slew rate that is required to go from the center to the periphery of the K-space. So our idea was to use small circle for that, for a lower slew rate. Also, in order to perform some under sampling in a very sparse way of the K-space, we have chosen to put this circle in a really random fashion. So we got a really nice sampling here. And again, a cost function, last one probably, with some acquired data, the low rank, the spectral, the spatial component, the forward model, the data fidelity term, and some regularization that is total generalized variation. This is actually a development of the TV, including now also the second order spatial derivative. And with that, we could acquire a high resolution FID MRSI at 17 3 millimeter isotropic in 9 minutes. And for this example here, we scanned a glioma patient where we see the tumor in flare, and we see clearly the drop in NA glutamate and the increase of signal in choline, glutamine and glycine. Let's see that are specific to the lesion. If you are also more interested about eccentric, I talked about it at the poster 13. And with that, I would like to conclude. I hope that I introduced how, with advanced acceleration techniques, we can perform a whole brain high resolution MRSI in less than 10 minutes. That would be compatible with the clinics. Of course, this is a really just a framework for reconstruction. We are really free to add the regularization, the prior knowledge, the low rank or not that we have in order to get the best result. But I think this inverse problem is really like a methodology, how to reconstruct MRSI data using the physical model. And also I recommend the reading of these three review articles. I think the two first were presented by Ovidiu, not the last one. That's where you might find some interesting information for accelerated MRSI as well. With that, I would like to thank you for your attention and thank my colleagues in collaboration for inspiring work. Thank you.
Thank you for the nice talk. Are there any questions from the audience?
Thanks, very clear talk. Problem I have is that denoising has a long and dishonorable history in NMR spectroscopy. And I'm sure that when you denoise it, it looks nicer, but I'm sure you've introduced some bias, and because every point in the FID is a sum of signal and noise, and the noise has random phase, so it either adds or subtracts to the signal. So I really don't see how you can separate signal from noise. I can see you can get rid of the noise in regions around the peaks, but the point which defines the top of the peak, it's still going to have noise on it. I don't see how you can
You mean?
How you can remove that?
You mean physiological noise? Or
No, NMR noise.
but MR noise is going to be really, incoherent. It's going to be fully different from one point to another. So the low rank really pick the, let's say, the coherent pattern inside your data set. If there is any redundancy between neighbor voxel, it's going to pick that. The one point you are correct is in a case would be, let's say, pathology that affect only one voxel with one peak that would appear only here. It would be taken for noise, and that would be that would be missed by a low rank approximation. But the low rank really aim to pick up the redundancy in your data set. And as soon as there is something redundant, it will be picked up. Noise is not redundant. I mean, it's fully random.
Okay, so if the noise is different on two adjacent pixels, then you've basically averaged it. So you've thrown away some spatial resolution. So how do you decide whether pixel A or pixel B has the correct value of NAA? So how can
we don't average it.
Well, effectively, you must be doing something like that because you're saying that the noise is not correlated, the signals are.
Yeah.
But I fail to see how.
Yeah, but it doesn't mean that we average them. I mean, when we pick the main component, for instance, that's I might just go back to the slides about the denoising for illustration. The main components most of the time always show some physiological. It doesn't mean that this map needs to be smooth. I mean, still the relationship between neighbors is kept. It's only that's really it's like a PCA in very high dimension. And really getting rid of something incoherent. Now, I agree with you, in case there is some signal that is incoherent, probably it will be thrown away. That's the only point.
Possibly.
Maybe to add to that quickly, like if you look at how our brain works, it does the same thing in a way. Like if you have a very noisy cow, for example, somewhere, there is signal, and what your brain does, it removes the noise for you in a way, and you see the pattern behind it. And that's denoising also. And there is redundancy because like the noise is random, but the image behind is not. And if you can pull them apart, it's like that can work.
Yeah, but I think you end up with a bias.
There is a bias. Yeah, you exchange bias for kind of precision. Yep, that's right. Maybe a quick question. Yeah.
Okay. Thank you.
Maybe a little bit larger noise.
Yep.
Some non-linearity in the reconstruction. They can miss that signal by this approach or.
Yeah. I mean, of course, the regularization aim to minimize or to avoid solution with some that are not regularized. So often, if there is, for instance, something look like noise, it might be again thrown away. But I mean, it's part of the method probably. If you would expect such a thing to happen, I would not recommend to use such a method when you are more interested in looking at the distribution of metabolites. Then you would be focusing on really the regions with a single voxel, for instance. But you're right. Well, okay. Yeah, no? Yep.
Yeah, go ahead.
Yep.
So how does the lowering approximation work with natural shapes that is like the perspective behavior of noising or
I'm not sure I can answer so straightforward, but of course we have to be very careful. If we keep too low rank, I mean, we're gonna miss some feature. Definitely, I mean, we have at least 5 measurable metabolites, even more, with different feature. So at least you need at least double the number of rank, because you expect to have a distribution of for each metabolite. So I would take that as a basis, and then basically you play it safe and you almost double it, and you checked whether your last component is noise or not, or contains some signal. And if it's only contains noise, that means that you're safe. If you still have some, that's the way I used to do that. If you still see some signal or some information in the last component of your low rank, means that that you should add some. Okay.
Okay, thank you. We will now move on to our non-educational talks, and the first of those will be given by Jack Hyton from King's College, London. And it will be about outlier rejection for fetal MRS.
Hello, I'm Jack Hyton. I'd like to talk to you about outlier rejection for MRS in the fetus and neonate, which is a baby that's just been born, which has been affected by motion. So cerebral fetal MRS has great potential to assess metabolic disorders in the developing brain before the patient has been born, and also to potentially detect harmful placental pathology. However, fetal motion and also the low SNR, where it can be 5 times worse than in an adult brain, the success rate of fetal MRS is low, and relatively few fetal MRS studies have been published. So the aim with outlier rejection is to retrospectively improve the data quality by selecting only the best repeats, by considering the spectral data only. And this is particularly applicable to fetal motion because, of course, it's not visible inside the uterus. And so therefore, you would have to integrate navigator images and also segment the fetus separately from the pregnant patient surrounding it in order to do motion correction. So the method proposed here is ranked phase shift outlier rejection. So outlier rejection is already offered in various MRS toolboxes. For example, in FIDA, each repeat spectra is compared against the medium from the whole scan. And if the difference exceeds a certain threshold, then that repeat is rejected. However, downsides of this is it depends on an arbitrary threshold. So this is weighted by how much the transient repeats vary from the median, but the user still has to input a parameter themselves, NSD, number of standard deviations. Another problem with this application is that if the fetus or neonate kicks itself into a different position halfway through the scan, then the median will be unlike the spectra acquired before and after that point. So therefore, the equation breaks down. So the approach that we've developed is to look at the phase shift of the residual water peak after water suppression, which is calculated by looking at the phase of each block of 4 repeats relative to the first 8 repeats. This might seem ironic because having a residual water peak is seen as a bad thing. However, when you're dealing with data with very low SNR, it may be the best place to look for artifacts which affects the whole spectrum. It's also looking at the phase is also insensitive to frequency drifts that are associated with scanner instability or cooling. The empirical inspiration for this approach came from experience in our perinatal department that when a neonate wakes up and starts crying or moving, then often the residual water peak changes from an absorption to a dispersion line shape, which can also be corrected using phase correction afterwards. However, it shows an indication of motion. It is also seen in fetal MRS scans when the baby hasn't been born yet, about 10% of the time, where the fetus suddenly changes position and you get this big shift in frequency shift or in the frequency or phase, corresponding with changes in the spectra as the voxel position has changed. So the mechanism behind this is that translational motion will cause the evolution of the phase during the MRS acquisition to change. So therefore that detects small fast motions, whereas a repositioning in the inhomogeneous B0 field without an update of the shimming will also cause a frequency and phase shift change. So the second novel part of this algorithm is doing ranked outlier rejection. So this works by ordering the blocks of repeats according to the residual water peak phase shift. And then in order of how severe the phase shift is, you add them to the average of the repeats and then apply your fit model in order to quantify your metabolites to each of the cumulative averages. Now, what you might expect is that as you add more data to the fit, the fit quality should increase because you have lower overall SNR. However, this is not the case when you're ranking your data according to the metric, because when you get to the end, you're adding corrupted data that's going to make your fit worse. So therefore, you should get a minima in the fit quality, which objectively shows where you should have your cutoff with your outlier rejection. So yes, the fit quality was the standard deviation of the 3 strongest metabolites you see in a fetal spectra, which is NAA, choline, and creatine. And a summary metric was found by weighting these with the concentrations of those metabolites. So as you can see in real examples here, when you look at the summary metric, there's quite an obvious cutoff point where adding more data will make it worse because of the motion artifacts. So in order to test this algorithm, 2 data sets were considered. The first one is a fetal data set, which was acquired on a 1.5 Tesla scanner. Often fetal scans are done with lower field strength because of SAR considerations and the width of the bore. So therefore, we have very noisy data. And in fact, 35 were selected out of 127 because most of them were just a complete noisy data set. The second data set that we considered was a preterm neonatal data set. So these were scans with a 3 Tesla Philips Achiever scanner, and the voxel, the MRS voxel, was positioned over the thalamus manually using an atlas image. These were separated into 2 groups. So all the neonates were born preterm, therefore before a gestational age of 37 weeks. However, 15 of them were scanned before 37 weeks, so preterm, and 35 were scanned after that threshold. So what we're trying to look at is how the metabolites recover or remain abnormal if a baby born before term, as it ages past the point when it's expected to have been born. So looking at the effect of the outlier rejection, the fit quality with no outlier rejection, with ranked phase shift outlier rejection, and using the FIDA function for comparison is shown here. The best performing NSD threshold was used for the FIDA function. And it's shown that there's a statistically significant improvement in fit quality using the ranked phase shift. And also with the neonatal data, the line width also improved with statistical significance. Whereas using the FID threshold of 3, which is standard, actually made the fit worse for the neonatal data set, at least. Looking in detail at one example, you can see how adding the ranked phase shift outlier rejection improves the separation of the peaks visually after fitting. And with the second data set, there was also this interesting result where the thalamus NAA in the neonate scans preterm was lower than those scans after term, which is what we might expect from previous literature as the compound recovers as the preterm baby ages. However, this only became statistically significant when ranked outlier rejection was applied. So to conclude, our proposed ranked phase shift outlier rejection algorithm significantly improved quantification of key metabolites in fetal and neonatal cohorts, and also does not require any arbitrary threshold to be chosen, which is a practical advantage. However, it's not the ideal solution because, of course, data does have to be rejected. The ideal would be to do prospective motion correction using navigator images integrated into the protocol, and perhaps using artificial intelligence segmentation in order to separate the fetus from the surrounding patient. Thank you for listening.
Thank you for this talk. Are there any questions from the audience? If not, like I would have one, like, did you also like, or do you think it would also work instead of using the phase to use the frequency? You said like that there is also a frequency.
Yes, so essentially we tried that. We looked at the frequency shift of the residual water peak and also its width and things like that. But the phase was the thing that most strongly caused the right correction with statistical significance.
Mm-hmm. Yeah.
I think that's remarkable data you've acquired from the fetal brain. That's great. Actually, just a couple of comments. So your NAA may not be recovery of NAA. It could just be the normal developmental trajectory of NAA, which increases with age during development.
Yes, of course, but also it's possible that being born preterm might affect that process. So of course I guess it's just an example application of this data set. Yeah.
Another thing which surprised me is you got fantastically narrow line widths, like 2 hertz or something, 2.5 hertz in the fetal brain. Is that right?
That's when you put all the averages together after the outlier rejection and also do the fit. So yes, obviously, if you look at individual transients, they look really noisy.
No, it's the water line width.
Oh, okay.
I mean, I'm surprised it's so narrow. So is that because there's no iron in the fetal brain broadening our line width, or?
I think a lot of it will be CSF. That'd be my guess partly.
Right. Yeah.
Yeah.
That makes sense.
If there are no other questions, then we will. Okay.
Thank you.
Also, when they're smaller, on the newborn, they have a better shield. So that's what at least the
Okay, then let's move on to our next talker, and I will hand over to my co-moderator.
Thank you so much. Next up is Rudy Rizzo from University of Bern. He's going to talk about torus simultaneous concentration and T2 mapping of brain metabolites by multi-echo spectroscopic imaging.
Well, good evening. Thank you to the organizer for inviting me, for having me today. And the first thing I wanted to do is convincing you why we are interested in concentrations and T2s. Well, we all know that concentrations, they do provide insight into functionality and pathophysiology. However, relaxation times could act as potential biomarkers carrying information about the cellular microenvironment. So the end outcome of our study would be carrying on a multiparametric MRS examination, providing both concentrations and specifically T2 maps. And to do so, we extended the single voxel technique into MRSI, which relies on acquiring multi-echoes in one shot. And that is twinned to a bidimensional simultaneous fitting. That is shown in single voxel to be fast and reliable compared to multiple multiple acquisitions. And it does not need any complex modeling as it would have been with fingerprinting. The method is rather simple. We take a Siemens semi-laser MRSI, 2D MRSI. We applied a metabolite cycle pulse early on. we run a weighted k-space acquisition, so there's no acceleration in the readout, but instead of acquiring a very long FID, we add 2 MAO pulses, so we add 2 refocusing pulses, and we chop into 3 chunks the FID. Therefore, we acquire 3 echoes, which are acquired in an FID fashion for the first one. The second and the third are partial echoes. And of course, the first 2 are quite undersampled. However, the trade-off we can build up eventually is having the shortest TE with a rather good intensity, SNR, and the late TEs with high resolution. So if you will, the speed up gain or the acceleration gain is threefold. We have a coherent scan. So among the 3 echoes, one would expect a coherent motion artifact if there would be one. However, we have to deal with low resolution for the first 2 acquisitions and gaps in the ADC. This is a pilot. The pilot has been run on 1 volunteer. This is the field of view. It's a 16x16 grade. We run 2 averages. It takes about 7 minutes. And the 3 echoes we've chosen are 35, 150 circa, and 280 milliseconds. And if we run the acquisition with a bandwidth of 4 kilohertz and 4,000 data points, you can see that the first 2 readouts are quite undersampled. We have rather 56 milliseconds or 200 data points, and the second one is double the size. The study design compares this multi-echo single shot acquisition, about 7 minutes, with the full acquisition, which is the multi-echo multi-shot, where we play the same sequence, but we either play 1 or 2 pulses, and then we acquire the full length of the FID. Here I report you a voxel in which you can compare the quality of the spectra. The multi-echo multi-shot is actually in high resolution, so you can pick all the resonances, quite nice. However, the multi-echo single shot is present in low resolution, and that's the consequence of having undersampled data. The trick is not considering those undersampled data alone, but putting them together in a simultaneous bidimensional fit and assuming void line shapes. To do so, we rearrange the batch into 2 dimensions. In one, you have chemical shift. In the other one, you have echo times. And the bidimensional fit works in this way. You fit the traditional line shape in 1 dimension, where you can see the assumption at the very bottom of void lines, where you have Gaussian broadening, mainly related to shim, and Lorentzian broadening, which is the measure of T2. The Lorentzian broadening can be extended on the 2nd dimension, and this builds up the bidimensional fit. 16 metabolites have been simulated and fit. The macromolecular background is simulated, and is considered uniform over the whole field of view. Prior knowledge is limited to 6 major metabolites, so we fit freely T2s for only 6 metabolites. Before showing you maps of concentrations and T2s, I wanted to give you a glimpse of how the spectra looks like. So I've chosen 3 voxels. One is in mixed tissue, one in gray, and the other one in white matters. In the 3 columns, you have the tissue types, and each of columns you have on the left the multi-echo multi-shot, so high resolve spectra, on the right low resolve spectra. I'm guiding you through some features we can spot, but first of all, you can see the fits are rather good. However, gray matter shows a lower peak of NAA rather than white matter, and one would expect the opposite. Second of all, we spotted the macromolecular background, or some residues of it, at 156 milliseconds. One would not expect the macromolecular background present at that long echo times. You can also see that the assumption of having uniform macromolecular background pattern is underperforming in all fits, and you can see some residues over there. But overall, the fit works. Diving into maps, here you can see a top row, concentration maps for 6 metabolites. On the bottom row, their relative Kramer-Rao lower bounds. Zooming into 2, glutamate and total creatine, I'm showing you now a plot on the top where we explore the field of view, pixel by pixel, and we have in 2 colors the multi-echo multi-shot and the multi-echo single shot. And you can see a good agreement between the estimate of the two approaches. If we look into camera lower bound and we correct for the scan time, we can see that the multi-echo single shot has a comparable accuracy to the multi-echo multi-shot. The most interesting slide could be, I guess, is this one about T2s, where you can see on the top row the 6 metabolites we've been fitting T2 freely, and the bottom their camera lower bounds. Again, if we compare the variation of the estimated values, they are quite in agreement between the two techniques. Comparing T2s with the literature is quite difficult because they do vary with the technique you run. So if you run a press or if you run a S-laser, they do vary, and there is quite an inhomogeneous data set. But we could say that the range overall agrees. And for what concerns the camera lower bounds, for instance, in creatine CH3 peak, the consistency between the two techniques is found. However, for glutamate, the uncertainty are quite high for this undersampling approach. So the conclusions are, we extended the single voxel technique to MRSI. We have tried it in vivo. Seems that there is a potential. We have shown that there is a comparable accuracy of estimates between multi-acquisition and single acquisition. The precision is increased, or inversely, one can rely on a threefold acceleration factor. We are now diving into inter-subject variability and stability. We also want to monitor the variability across different tissue types, and for that we need segmentation tool. And I hope William can help me on that. And we're also working on an extension to prostate with a specific tailored sequence. With that, I would like to thank you, the group in Radboud, especially Elena, that helped me and supported me quite quite a lot during 6 months, and especially Roland for the MRM group in Bern. And thank you all for following me. And if you have any inputs or questions or feedbacks, please feel free to help me. Help us.
Great job. Are there any questions? Here, let me just.
Hi, nice presentation. I'm sorry, I might have missed this, but you have this 2D approach. I get that. You have like different echo times. That's one of the dimensions, right? So how do you prepare your basis set for this fitting approach or for your 2D approach? Do you basically just say you have a different echo time, you ignore the additional 180-degree pulses, or do you consider all of those?
Yeah. The additional pulses are considered. Yeah. So the
Thanks.
NAA undergoes another multiple.
I see.
Yeah.
Thank you.
You're very welcome.
Are there any other questions? Coming down.
Yeah, just really quick and just following up on Christoph. So the optimization is actually done in the time domain. So you do a time domain fit? Or was
No,
that frequency
I
domain
show
fit?
the model in time domain, I know, but I do fit in frequency domain, so I can select the range.
How does that then with truncation artifacts? Like how does that?
The basis set is truncated as well and zero field, so you actually
Oh,
have
so they show up right
match.
away. Yeah.
Yeah.
Have you considered fitting in the time domain and actually save you some trouble? I
So
don't know.
fitting in, you would have this like step going down, so I don't know how it would work. And then I rather like to select the frequency range to fit personally.
Gotcha. Interesting. Thanks.
Are there any other questions? Okay.
So these approaches would require a long TR, right? Because of the number of pulses that you use, it would only work with
1400.
And are you working under almost fully T1 relax conditions, or is the TRs that you use is
TR is very long, 14, 16, depends also on the pulse duration because
is it 14, 16 or what?
100 milliseconds.
14, 16, 100 milliseconds. Okay.
Also, it depends because the pulses might be longer or shorter according to the head.
And so the last pulses that you add at the end, they are, so you had some sequence where you use semi-laser, and then so you use this adiabatically focusing pulses, and then the ones that you added are just normal. And you use gradients during the pulse, or are they slice selective or unselective?
They are slice selective, and they're slightly bigger, 1.5. So they do refocus 1.5. A slice that is 1.5 bigger than what has been selected before. So in such a way, we think we can ensure a refocus of everything that has been excited before.
So those pulses just select axial, so you have gradient in the slight direction, or
yeah, two directions orthogonal. We cannot refocus all the three of them, but two. Yeah.
Okay.
Thank you so much for a great presentation, and we're moving on.
Thank you.
Next up is Brian Alves from EPFL.
Hello, hello. Oh, perfect. So, good evening or good afternoon. We're on the border right now. Thank you for being present, and thank you for the opportunity to present my work on the Marchenko-Pasteur PCA and low-rank TGV denoising in proton FID MRSI data at 14.1 Tesla, application and validation. At Outlier field in the clinical setting, proton FID MRSI acquisition are increasingly used, especially for neurodegenerative pathologies investigations on glioma, sclerosis, epilepsy, to name a few. However, in the preclinical setting, this acquisition is not widely applied due to problems with low SNR, small nominal voxel, lack of process for huge data sets. And as we've seen with Ovidio and Antoine, there is a higher need for spatial resolution and speed. And among those methods, we have found that there are post-processing denoising processes that we can use. And it's been the aim of my work. It's to enhance the implementation, the feasibility, the routine usage of two denoising technique, the low-rank TGV and the Marchenko-Pasteur on preclinical 14.1 Tesla fast proton FID MRSI dataset. The data was acquired on a 14.1 Bruker magnet with a homemade transmit and receive quadrature surface coil, and using a T2 weighted turbo-rare MRI slices, we position our MRSI slice at the center of the hippocampus. With a thickness of 2 millimeters, a field of view of 24 by 24 millimeters squared, and a matrix size of 31 by 31, we reach a nominal voxel size of 0.77 times 0.77 times 2 millimeter cubed. We have 1,024 spectral data points done on 1 average and 2 average acquisition, with an echo time of 1.3 millisecond, a repetition time of 830 milliseconds, for a total time of 13 minutes. But let's talk about the denoising process. We have the raw reconstructed MRSI data acquired directly from the magnet in the format of a 31 by 31 matrix. And first, we do some pre-processing. We phase correct the spectra, and we do some lipid suppression using a metabolite lipid orthogonality approach by using a separation of a brain mask and a skull mask, with basically using a power mask, which allows us to, while using the water signal, to differentiate the subcranial lipids by simply analyzing the power of each voxel and the average power of our signal. So now we have the pre-processed MRSI data. We triplicate this data by applying the LR-TGV reconstruction, and then the MP-PCA reconstruction, and the data non-denoised. As a result, we obtain 3 matrices of 31 by 31 voxel containing our signal, which are both denoised and non-denoised. So I will quickly go over 2 denoising techniques, which are the star of the show, obviously. We have the LR-TGV technique, which is the low-rank total generalized variation method, which is a method applied in clinical MRSI. And the assumption is that there is a finite number of metabolites independent of their spatial location. So to paraphrase what Antoine has said, it's the partial separability of spatial temporal modes, which allows us to separate to our signal data into two matrices, one that has the spatial information, the U matrix, and the other that has the spectral information, which is the V matrix. Those two matrices are linked with the rank component, which is something that we can set manually during our reconstruction. And the total generalized variation regularization is a constraint that we add to the cost function, which allows us to avoid staircasing effect by applying it on the spatial component. Now, the Marchenko-Pasteur principal component analysis is a method that has been implemented on diffusion and functional MRI that works with the Marchenko-Pasteur distribution and fits them to the eigenvalues of the covariance matrix, meaning that we get a certain number of noise eigenvalues. And the goal of this method is to find the P signal-related eigenvalues and to use them as we reconstruct our matrix using singular value decomposition. So we have our triplet of data. Now we quantify them using the LC model. The basis set was obtained from simulation in NMR scope B, and we fit the spectra for the voxels of interest. From them, we gather the concentration values as computed by LC model using total creatine as reference, and we store them into a 31 by 31 matrix to represent the MRSI slice. And there are other values that we gather from the LC model, which is the signal to noise ratio and the full width half maximum. And those are used as a automatic quality control as a way to filter out spectra that have low SNR and the highest full width half maximum. So we have used a condition, which you can see here, which is 0.75 times the average SNR computed on the non-denoised data and 1.25 times the average full width half maximum value. From there, we get concentration maps like this. So we see the concentration maps for inositol, NAA, and glutamate. For non-denoised, MP-PCA denoised, and LR-TGV denoised. And one of the results that we have seen is that we see an increased coverage of the concentration values due to a higher value of accepted voxel, which is expected since with denoising we observe a higher SNR, and thus we bypass the threshold that we have set for the quality control of our data. And this becomes more obvious when we compute the apparent SNR for this time the denoised data sets, and we observe that the average SNR is generally doubled when we compare with the non-denoised data, and we observe an increase of 10% in the coverage of voxels. As a comparison, we did the same exercise for two averages, non-denoised, just to have a point of observation. And we saw that the percentage of accepted voxel is perhaps a bit higher when applying denoising. So now we wanted to focus on the values of concentration. And if we do keep some information concerning the average concentration on certain brain regions. And for that, we have decided to compute the average value of the concentration of metabolites in two regions, a mix of cortex and striatum in purple and hippocampus in blue. And we have decided to compute an average value over four rats. And what we have observed is that the standard deviation is reduced each time when applying the denoising. And perhaps more importantly, there are no statistical difference between the methods, meaning that the average concentration computed with non-denoising stays relatively the same, at least has no statistical difference compared to what we obtain after denoising. But there's also the behavior between the brain regions that is interesting, and that's something that we've also investigated. And what we have observed is that the behavior compared to what we have found through literature stays relatively similar to what we have after and before denoising, meaning that at least the regional distribution for those three metabolites remains the same even after denoising. So to summarize, we have done a successful implementation and routine usage of those two denoising techniques on fast proton FID MRSI datasets at 14.1 Tesla. We have seen an increased coverage and average SNR on the concentration maps when using the denoising techniques, and no significant difference in average concentration between the three methods has been observed, while we also kept the brain regional distribution for glutamine, inositol, and glutamate. The perspective for further studies is to use, of course, Monte Carlo simulation as a ground truth, especially for the case of lower concentrated metabolites, as we want to observe if we do indeed keep the concentration after denoising, and to avoid the addition of random information. So I would like to thank you all for your attention, and thank my group for all their help and work like this. And if you have any question, please feel free to ask. Thank you very much.
Thank you so much for your presentation. Are there any questions?
Probably I have a little bit silly question, but can you consider LC model fitting like ideal denoising?
Like sorry,
the fitting LC model, if it is not, if it can be considered as an ideal way of denoising of your data spectra.
No, I don't think so.
Because you are getting spectra without noise. Oh, sorry.
Any other questions? I'm not sure. Do you want to address it again?
Well, in a way, if you fit data, that can be considered as denoising also in a way, right? Like after that, your data is noiseless, right? Or your signal is noiseless, and like I think Ivan has a point, like the fitting, how it's done in LC model with the, I think it's the root mean square error minimization, or L2, I think, is kind of in many cases the optimal way how you fit just individual spectra. You don't take into account any spatial information, but for just one voxel or one spectrum, it's the optimal way to kind of fit data and therefore denoise in a way. I would have one more question. Like you do this like lipid regularization, but at the same time you use I missed it, laser or press box or something, right? So I've never seen that that you can use lipid regularization plus like you have very little signal from the lipids due to the laser or press localization, right?
Yeah, but we still do have a bit in our data, like some residual lipids actually.
Okay, that's
So
enough to kind of.
It's to create a bit of a leakage on the data. Yeah,
But they are not kind of changed in a way that they so that you can still subtract them from the brain, basically.
Yes, exactly.
Okay. But how do you get the rectangular box? Okay. Okay. Then. Okay. Yeah. Okay. Thank you.
Thank you so much. We're moving on to the next speaker. Next up is Ipek Özdemir from Johns Hopkins.
Hello, everyone. That's the last talk. We are almost there. Please bear with me. And we are going to the other side of the water resonance. It will be very exciting. Yeah, thank you for a nice introduction. My name is Ipek, and today I'll be talking about downfield proton MRSI in the human brain with extended spatial coverage.
In proton MRS of human brain, signals occur both upfield and downfield from the water resonance, whereas upfield resonances originate from protons bound to saturated or unsaturated carbon atoms. The downfield resonances typically originate from aromatic or exchangeable groups. Oops, what I wrote. A number of compounds of biomedical interest resonate in the downfield region of the brain. For example, homocarnosine at 7.08 and 8.08 ppm, N-acetyl aspartate at 7.9 ppm, amide from mobile proteins at 8.1 ppm. However, the detection of these resonances are challenging due to the low singleton noise ratio. In conventional brain MRS, water is typically suppressed by presaturation. And as a result, the downfield resonances are usually suppressed too, because many of them are rapidly saturated due to either chemical exchange or cross-relaxation with the saturated water magnetization. The downfield resonances also typically have quite short T2 relaxation times, so they are also attenuated when data are acquired at long echo time. So to be able to detect downfield resonances with an increased SNR, one should avoid pulse sequences that saturate the water magnetization. Furthermore, the concept of relaxation enhancement can also be used to improve SNR of downfield resonances, making use of exchange from the large pool of fully relaxed water magnetization to rapidly restore the downfield resonances to equilibrium magnetization after excitation and detection. The peaks shown here on the right-hand side, these downfield spectra are commonly observed in the literature, and most of the studies about downfield resonances in the human brain have used single voxel localization methods, but this is a sample from a downfield MRSI experiment. Recently, an MRSI technique capable of mapping downfield proton resonances in the human brain was implemented by our group. A spectral spatial excitation and frequency selective refocusing scheme in combination with two-dimensional phase encoding was developed to map downfield resonances without any perturbation of the water magnetization. The numerical calculations of the excitation and refocusing profiles overlaid on a typical brain spectrum on the right-hand side, showing that the excitation pulse is shifted from water resonance to avoid any perturbation on water magnetization. Using an echo time of 22 milliseconds and voxel size of 10 by 10 by 15 millimeter, 9 downfield resonances were mapped in a single slice MRSI experiment for the first time at 3 Tesla. And left bottom image depicts the slice location used for these MRSI experiments. And in this work, any water suppression was avoided and relaxation enhancement effect was used for high SNR. And improved SNR has been demonstrated with and without water suppression on the MRSI data as shown on the right bottom corner. Based on these previous efforts, in this current study, our objective is to develop a three-dimensional downfield MRSI protocol with near full brain coverage and high spatial resolution at 3 Tesla. I just realized I still have my, I hope you can hear me. Okay, let's continue. Okay, for this, we implemented a three-dimensional circularly phase-encoded version of the previously developed two-dimensional MRSI sequence with 1331 spectral spatial excitation and frequency selective refocusing. And we have tested this pulse sequence in 4 normal human volunteers using a Philips 3 Tesla scanner equipped with a 32-channel receive head coil. We acquired data from 120 millimeter slab in 8 slices. After the localizer images were collected, we acquired three-dimensional FID MRSI data without any water suppression, and this data was used as reference in assay model fitting later on. Here on the left-hand side, I'm showing you the common parameters used for water and downfield scans. Our field of view dimensions were 180 by 200 by 120 millimeters with a nominal voxel size of 7 by 7 by 15 millimeters. We applied second order fast map shimming. Water scan took 11 minutes and 14 seconds. For downfield data collection, we had a TR of 287 milliseconds, and the echo time was 22 milliseconds. The 1331 pulse delay was set to give maximum excitation at 7.4 ppm. The frequency selective refocusing was realized with a 180-degree sync Gaussian pulse at 7.8 ppm, with a duration of 11 milliseconds and 400 Hertz bandwidth. And three-dimensional downfield MRSI data was collected in 22 minutes and 42 seconds. Here's an overview of the post-processing pipeline. We used the basis sets for the downfield peaks created in our previous study based on the literature for the frequency shifts. And because of the uncertainties in the peak assignments, the downfield spectra were fit as a collection of individual Gaussian peaks. Okay, sorry. Then we fit the downfield data using SE model software with the water referencing slice by slice. The SE model baseline spline stiffness parameter was set to 5. And based on the fit values created by SE model relative to the water scan, we created the individual metabolite maps for each subject, showing the spatial distribution of the downfield resonances, as well as summary statistic from 4 subjects for 3 different brain regions at the peaks 7.9 and 8.1 ppm. The images on the right-hand side demonstrate how we performed voxel selections in different brain regions. And the spectra from those representative voxels show similar downfield resonances, NAA and MI groups being the largest peaks at 7.9 and 8.1 ppm. These results are in line with the previous downfield spectral profiles published using single voxel localization techniques. We selected voxels from anterior cingulate cortex, in short ACC, from centrum semiovale, in short CSO, and from dorsolateral prefrontal cortex, in short DLPFC. The metabolite maps from 8.1 and 7.9 ppm peaks are depicted in the top row next to the slices with voxel locations. And some signal dropout from most inferior and superior slices were visible due to slab excitation profile. Also, some artifactual hyper and hypointensity was observed, particularly in inferior slices due to field inhomogeneity, in particular residual water signal when V0 inhomogeneity shifts the water peak into frequency ranges excited by the 1331 pulse, which is 7.4 ppm. And these images are preliminary, and they could be improved in post-processing. In the bottom row, the selected downfield spectra from sample voxels in ACC, CSO, and DLPFC is shown. Similar pattern on the downfield resonances can be seen in all 3 brain regions. In the summary statistics, it can be seen that although the SE model estimations for different brain regions are not significantly different from each other, a somewhat large dispersion was observed within individual brain regions. And all selected voxels were quantified with a CRLB below 15% for both resonances. In conclusion, downfield MRSI with 3-dimensional coverage is achievable at 3 Tesla in a clinically feasible scan time. There are a lot of advantages in downfield MRSI. For example, we have the ability to use short TR, and there is no need for additional water or lipid suppression. The downfield resonances are slightly less sensitive to field inhomogeneity since they have broader intrinsic line widths compared to upfield. And downfield MRSI may give also specific metabolic information on slowly exchanging molecules that is complementary to that observed using CEST MRI, which is more sensitive for intermediate exchange rates. So with that, I would like to acknowledge the funding agencies and thank to everyone who supported this project, and thank you for listening.
Thank you so much, Fetal, for an amazing presentation. Are there any questions?
Yeah.
Microphones are not working, so you need to be really loud.
Okay,
Oh,
it's working. Thank you. Very interesting. I'd like to know, for you use this specific excitation pulse, does it introduce any chemical shift displacement artifacts?
It might be. I think in the maps, I see some spurious pixels, and that might be actually from that reason. You know, the not inhomogeneity, if it is shifted to excitation area 7.4, the 7.9 maps are showing this information, I guess,
Okay.
or artifacts, not information.
Yeah, thank you.
Is there any other questions? Yes. Want to grab the microphone?
Well, I can shout. So can you see the creatine resonance?
Creatine resonances.
Could be a creatine and phosphocreatine from the other field.
Okay, if you remind me the ppms, I can.
I can't remember.
Yeah. Well, I'm not sure about if I'm seeing them or not. I can tell you I have 9 maps actually created from 6.8, which is a stable signal, glutamate, I guess. And 7.9 was very stable because it's slowly exchanging. And 8.1 was also very stable.
Finally, it worked and they took it away. One comment is you mentioned relaxation enhancement effect, but I think you should stress that this is just for exchangeable peaks. I mean, your NAA, for example, does not really profit from that.
Right.
The exchange is far too slow. So you're really stressing out the exchangeable peaks, 8.2, 8.4, 6.8, whatever.
Oh, okay. Yeah.
Just to clarify that. The other thing I was wondering, you have this chemical shift selective refocusing instead of a slice selective refocusing. Why don't you just use a normal slice selection and you can pre-invert the water? And that's what we did in single voxel. I mean, you don't have to do this chemical shift selective inversion because you're not exciting the water if you can just refocus.
I think if I'm wrong, please, Mikael, correct me. In the previous work, they tried first this 1331 pulse, but then they got a error. First
It's just
level
you have too
gradient
much
error.
water excitation. That one.
Yeah. So that was the need for refocusing again. Thank you for the question.